right, I think
sort of hold off.
seeing the bottom here. Good afternoon, good evening, good morning, wherever you are.
Why don't you push out a little?
This is
just
a
a
live video
sort of under me
broadcast
there.
from the NOAA ship Okeanos Explorer.
Yeah.
And it's Deep Discover ROV.
Just have to
We are
work to fight it.
currently at approximately 2,235 meters down on a ridge extending from Beatty
so it looks like
to
it's
Seamount.
blowing you
This is inside the wake
west
unit
a little
of
bit,
the Pacific
maybe?
Remote Islands Marine National Monument.
Pretty hard here.
We have
Look at the
bottom
return
in
on your
sight,
sonar.
and we'll be getting there shortly.
I've got a little feature possibly here, maybe.
So my name is
looks like I have the ridge
Chris
off to
Kelly.
my
I'm with
starboard,
the University of
and your
Hawaii
sonar would agree. So
Oceanography Department.
we're landed just
And
a little
next to me, we've
little
got
bit west here, so we can
Jasper Conter.
acquire that,
I am with
get up
the,
back to the top of
Department
the
Geology
ridge,
and Geophysics
then
at
track
the
along
University
it,
of Hawaii.
maybe. So
And
get
we
caught up to
are
you a little
hoping
here.
to see both some nice geology and biology on this dive.
There you go.
We're seeing our first glimpses of the seafloor at this point.
Okay.
We're getting settled here and start looking in a
An
minute
easy stop for a second.
at what's in
Looks like
view.
you got pushed out a little ways. Giving you little tugs. Why don't you back up just a hair?
Copy.
I'll keep coming down. Keep on, keeping on. Okay. I'm going to hold off for a second here. You keep backing. And we're going to line up, maybe cheat it a little and do northwest, but we want to make our way, or northeast, but we want to make our way east a little bit before we start headed up the ridge, I think.
So we saw a couple
So
of
I'll
animals
start
as we were
steering
coming
that
down.
general
Look
direction.
like a couple of sponges, and I think that's what we're seeing right now in the upper left corner.
And you just
But
line yourself up
we're
best you
not
can on the
seeing a
hill.
huge number of different animals at the moment. Perhaps we'll see some more.
Yeah, so I have the ridge basically due west of me, but you've got it sort of northwest or northeast. I have it due east of me. You have it sort of northeast. So
what would you describe the substrate as being, Jasper?
Oh, there you go. You're lined up.
This is
Okay.
much more
So
solid
we might make a
rock
short
than
hop
what we
east,
had on, our
and I guess
first dive.
actually
We have,
HIPAC sort of agrees.
Longer stretched
So to get
out
Serios in the middle of the ridge, we
things,
might want to make a 20 or 30
underwater
jump
features.
east.
Usually our pillow lava,
Probably
so
30
sometimes
meters,
with sheet
and
flows on
maybe
the
we
bottom. These are obviously
maybe we
longer features,
cheat
so
a
these could
little
be
towards
tubes,
the north,
these
but
could
it's
be,
the ridge is already heading
the edges
northeast,
of
so we're
flows.
just going to keep paralleling along. So why don't
We're
we just
getting
head
some
east?
animals in view
Does
now.
that sound like a plan to you?
Yeah, we're getting a bunch of things.
Okay.
We're getting a really nice
I'm pretty
glass
much lined up
sponge
on
here at the
east,
bottom
so
right. And there's
I'll
a
come
dead
down a
sponge
little
up
bit
in the
more,
upper right corner.
and
We
you
also seem
can
to have some unbranched,
start to explore, and
possibly
Nav and I
cnidarians,
will discuss a
if
move.
they're still alive, that are scattered around the bottom between the two.
Yeah.
So guess we just needed to
So
get
just one tap to
a little bit
re-enable.
closer
There
to
you
the
go.
bottom before some of the stuff started showing up.
And righty-tighty keeps you down, right? Lefty-loosey pushes you off. And you still have auto heading locked in, just in case. You can leave it if you want it. You don't have to have it at this point if you don't. Let me make sure the swing arm I put back outboard. Lower swing arm. Outboard, there we go. Okay. I think I'm set with hydraulics, so I will power them down. Okay. Hydraulics disabled.
Copy that. Yeah, I'm not really sure what those little
Come down a little more with you.
stem things are. I think this large white sponge in front of us, which I assume we're going to zoom in a minute once they get the ROV
Okay, Nav.
squared away. is
Go ahead.
well, it's definitely
Go ahead,
a
co-pilot.
glass sponge.
How are we,
It could be in
how are we
the,
looking on your end?
family Eurydidae, but I'm
Good.
not
I,
absolutely
you know what?
certain at
I'm
this
trying to
point.
drop a target, and it's dropping
These
the target on the
sort
ship.
Of,
Okay, so
don't-
stubbles are
go into vessels on the survey bar.
Not sure what those are
up
at
top.
the moment, but we'll undoubtedly get a
I
better
see
look.
vessels is one of the pull down
also got
areas.
some
Yeah, and
dead-
then highlight
Appear to
ROV.
be dead Cnidarian
Hit
or
the little main
I
vessel
put
check
dead
in the
coral,
top right.
excuse me, or sponge stalks
Yeah.
on the bottom
Apply.
as well. Those are those things that look like dead tree branches.
All right,
I
much better.
I don't know if that worked.
Yeah, it did.
Oh, it did. Okay. I didn't know if you had to hit apply or not.
Okay, so I heard you
Looks like
discussing
some
a
of these
pilot,
guys actually
talking
do
about
have a little bit of
move due
branching
east
up
about
in the
30
top
meters.
Yeah,
Uh,
yeah, I think
I mean,
they're
how do
gonna
you
turn
feel
out to
about
be,
that? Due east move.
octocorals or Cnidarian or
I
something
see
there, but,
due east. The top of the ridge is
Hard to
maybe
really
40 or
identify
50
them
meters
yet.
away. We'd have to move almost southeast to get to, if we wanted to make a short hop just to get right on top.
Yeah,
But if you measure,
right,
if you
a little
measure
bit more.
from Sirius to the middle of the ridge due east, what do you get roughly? Yeah, middle of this blob. Does that say
it's in nautical miles for some
nautical
reason.
miles? Yeah.
It was in meters.
So you have to change the settings to
Oh, it's range.
no.
Oh, yeah. Oh, it's hidden behind your bar. So if you take this window and drag it down a little bit, then.
Oh, there you go.
Shows
Distance
Thanks, guys.
meters. Perfect.
some lasers on if you want. Go by it.
It's about 40 meters to the middle of the ridge.
Okay. Have you talked to science yet at all about
Touch
this
base
landing?
about moving up
Okay.
the ridge.
Okay. We can let one of you guys can let them know that we're here. And
Sure.
have you got your Z-bias settled, basically?
Yeah, it's pretty good.
All right. Well, you can tell them we're ready to
Start We're gonna move due east about 40 meters, then we'll start going up the hill.
consult with them a little. Yeah.
Watch lead pilot.
Roger, pilot, go ahead.
Yeah, we're just getting settled here, and yeah, we're ready to start looking at things for you, and then maybe get a move underway in the next couple minutes.
Be sure, terrific. And I guess first couple of targets, maybe get a close up of that large sponge, and then let's take a look and try to figure out what these little leggy things are.
Copy that. We'll get the large one here.
So we'll hold out on that move for just a second till he sorts out the sponge and the leggy
Okay.
things.
Copy that.
Okay. No, that's okay. I thought I was doing something wrong, so, all right. No, so as we're getting squared away, those of you who are new to the Okeanos dives, welcome. You're going to see two little orange dots that are on the sponge right now. These are the laser scale, and they're separated by 10 centimeters. It gives us an idea of sizes of things on the bottom. The lasers, of course, are not anywhere near powerful to do any type of damage to the animals themselves. They just,
Good, Bridge.
they're like little lights that allow us to get measurements. So Beatty Seamount,
Bringing your
or
scale
Beatty,
in a little.
geode, if you will.
Copy
It's
that,
a
and
flat
just so you know,
top
the
seamount,
ROVs are on the
sometimes
bottom,
called a
and our
table
bailout
mount.
direction would be
And we're not actually
around
on
100
the summit,
degrees,
the flat
110
part.
degrees.
We're actually on a ridge that's extending out from the
Copy.
summit toward the southwest.
Nav co-pilot.
We often like to dive on
Very
ridges
moment the
because,
ridge is at
they
110
tend
degrees.
to be sites where we
So
can
I
get
would say the
a fair
bailout
amount
would be
of,
opposite of
corals
that.
and sponges.
Till at least we
These
get on the
communities
bridge.
seem to like ridges,
Okay, got it.
perhaps because ridges tend
Right.
to, if they're oriented correctly, they
We'd
tend
want to
to
bail out
accelerate
more like
the bottom current flow.
285 or something.
filter feeding organisms.
Oh, okay.
like
Once we
corals
get
and
once we
sponges,
get over the ridge,
seem
then
to
we'll go.
prefer
Since we're on the ridge
Yeah,
right
sure,
now in
high
this
flow
area, if
environments.
something was to bad happen in 2 seconds, we would want to actually go around 285. So you should relay
285, sure.
that, or you can just say west. Might be easier to remember.
I think we got a black
Bridge is
coral
nav.
over here to the right too, which is a little different. That sort of brownish coral that's right on the right
Till we
edge.
get over this ridge, I'll let
So
you know.
maybe we'll
Our bailout direction will be due west.
Take a peek at that. We've actually got a bunch of stuff going on here, a bunch of different things.
That's correct. We're on the other side of the ridge. Once we get to the top, we'll adjust accordingly, but I'll let you know.
Okay, video. We'll try to settle in here real quick, and then we'll zoom in on this
Copy
guy.
that. It should just be a
Roger that.
short hop here.
I'll let you know in just a second here. Just get a little better perch.
So in the
Okay, video,
meantime,
come in a little
for
bit.
those that
Go by, you can
have
take
been
my
looking at the
rotator.
screen,
Do
the
video
rocks,
once,
the
but I
black
guess
rocks,
I'll try to hold it here. I got a little bit
look
current
very
in my
dark,
face.
mostly
I'm gonna
because
try
they're
to
covered in manganese crust.
hold this.
As was mentioned in the chat room already,
Okay.
this is
I have rotator.
fairly
Video, feel free to
thickly
come in
crusted
now.
over, and
Get this guy.
Zooming.
we can still sort of see some of the structures that are underneath, sort of giving the larger scale structure.
Just gonna hold
But,
this partial for a second.
At a smaller scale, the little bumps are all manganese crust, probably.
So this particular sponge,
Okay, let's take a bit more
I do
zoom.
think it's in family Irididae. That's a decent guess. This is what we refer to as a hard sponge, in that it isn't soft like a kitchen sponge. This particular family and the species that are in this family are quite brittle, and this is because their skeletal parts, which are called spicules, are fused together to form a very rigid
Damn right.
framework, and they're also fairly thin-walled, as you can see.
Not for a
They're
wide
fairly
shot.
translucent, so when you try to grab it, they can just sort of break up into pieces rather than just squish. So this is a beautiful sponge. We have a very difficult
Just the
time
lower swing arm
trying
ever
to
so
age
slightly.
these things, but this one certainly looks like it's pretty old. If you look at the
Take
size
that
of it,
flare out of there.
maybe 50 centimeters across.
Okay, video's clear.
Nice.
Full wide.
Watch the pilot.
Let your pilot go ahead.
Would you like to look at the black coral, or what's
Yeah.
next?
Yeah, so let's go ahead and look at that branchy brown thing that's on to the right of the screen right now
Copy
first, and then maybe that sort of orange one above that, and then we'll need to get a couple of these white ones before moving up.
Sounds good.
Did the flare go away?
I could not tell.
Lens flare? Can't tell either.
Okay, video, you can zoom in on this guy. I might need to get a different angle because he's coming right at me, but
Roger.
we'll give it a shot.
Let's see what we can do.
Okay, so this is
Okay,
definitely a
partial for a
black
second.
coral, and let's see if Tina Molitsova is on or Scott France. They don't seem to be on right now.
Alright, going for a bit more zoom.
My guess would be something like Trissopathes, but I'm not absolutely
Want me to
certain.
tilt you down a touch?
Yeah.
Down.
Perfect.
Another option could be Heteropathes, but
Okay, I'll come
I
out for
suspect that
establishing.
Trissopathes might have a higher chance of being correct. I'm looking through the Okeanos Animal Guide. It's an online guide. I happen to have it on my iPad
Okay,
here.
clear.
Nice.
To
And then let's go for that orange
try
guy.
to get a-
Hold on, if you can take my rotator again.
try to
And tilt
confirm
up.
what it looks like.
Thank you. And if he's too far away out of the light pool, just let me know video. But
It looks like it'll be just great.
okay, excellent.
Zooming.
in the center here.
Okay, this is another type of black coral, and
Okay,
I'm pretty
I'm
sure
gonna
this
frame it
is in the genus
first.
Stauropathes. These are really beautiful corals. They tend to be almost translucent, glistening gold or orange, and they have sort of sub-branching going on, which you can clearly
Right,
see.
I'm
You've
gonna
got a
take a
main
bit more
stem,
zoom.
and then you have some lateral branches coming off
That's
it.
full zoom, so I'll just hold
And
that for a
Scott's
second or
on
two.
now. Great. So the cavalry has arrived. So Scott works on these, and so this is terrific. He'll give us some help with this. So Scott believes it's Stauropathes also. Scott France is at the University of Louisiana, Lafayette. And he specializes in
Alright,
black
clear.
corals as well
Thank you,
as
Lydia. I think they wanted to
chrysogorgid
see-
octocorals
Quick
and
frame or two with the
acidid
lasers inside.
octocorals, as
Okay,
well as
good.
an awful lot of other things.
So what leggy
But I
things?
wonder
oh,
if we could zoom some of
maybe
these,
this-
just a
oh,
couple
this
of
good
these
one
white
here.
colonies. I think there's several different things here.
Okay.
Yeah,
Roger.
Just
sounds good, Video. You can come in on this guy. Yeah, tilt down a little bit.
so I think this is some type of primnoid, but as they get a little bit closer,
Full zoom.
yeah, I'm pretty sure it is a primnoid, but what type? I have no idea. It's sort of these two-branch primnoids. I don't recall ever seeing anything like that before. Primnoids are a type of octocoral that are quite common in deep water. It's one of the more common members of the deep water cnidarian fauna. And looks like Asako Matsumoto is joining us. She's from the Chiba Institute in Japan. She concurs that this is a primnoid, as does Scott. So, you know, all three of us are in unison here that at least we can go to family on this one.
Video's clear on that one.
Yeah, I have a feeling that a lot of these white ones are
All right. If
primnoids
there's not much
too.
more to get here, we can start
Um,
moving.
Sounds good.
Okay.
Okay, so Pilot, maybe as we move up slope, we can stick to this more consolidated substrate. I have a feeling we're going to see more corals if we can. You know, as soon as we got over in this area, we started to see quite a lot of things
Yeah.
growing on the bottom. So, yeah.
Yeah, Coppola, I think the direction of movement is going to be pretty close to east. Am I correct?
Yeah, I'm lined up on it.
Okay.
Scott's wondering whether the two-branched ones get more branches and develop into the four-branched ones we're seeing.
Yeah, possibly. These are
So
all the
pilots,
same
do you
age.
wanna,
This is quite a
ship move put
settlement
in?
event that took place here. You know, I don't
Up
know.
to
I mean,
pilot. I'm ready.
It's unusual. They're so
He's getting
they're so
stretched
small,
out pretty
and
good.
they're quite abundant.
Yeah, we got me at 23. Oh, yeah. Well, actually, pilot.
Roger, pilot. Go ahead.
Yeah, we're ready for I think the first move here. We're gonna go in. I'll turn my head just so you have an idea of where we're going.
Okay.
But to get onto the ridge, we're gonna make a move due east. So, which is my current heading, pretty close to it.
Great. Thank you. So there you can see, you might see a very sort of tall sponge
So
that's
pilots,
off
I
to
got
the
about
left.
40
and this
meters
is
to
in
the top of
genus
the ridge.
Tritopleura, I believe. This is a,
Yep.
a fairly common sponge.
40
in
meters
deeper
due east
water.
sound good at 0.2 knots?
At least we can go to genus.
40
And
meters is
there's
good with
also
me,
a
pilot.
little small
Yeah,
sponge
that's good.
over in the lower
Okay.
left corner, and I think this is something that's called Hyalinema.
Regis Nev?
It's, I sort of think of them as the
Like to put in a
pot-bellied
ship
sponge
move?
because they tend to look like they're a little pot on the bottom.
Like do 40 meters due east at 0.2 knots.
Is that a fish in the
like-
back
Oh
there?
yeah,
in the
there's a
background.
fish in the background. It's like
Near
one
the
of
top
our.
of the screen.
It's like one of our
That is correct.
our friendly cutthroat eels.
Yeah.
But you can try to zoom real quick, but
Roger.
she might be out of the
No
light
worries.
pool.
We'll see what we can do.
It's going to be a in the family Synaptobranchidae, or that is the cutthroat eel family.
Nav copies.
He's getting away from us.
He's getting away.
Yeah, you can pull out video,
Roger.
and we'll come down and look at some stuff close by here.
Pilot
We
ship
saw a
move
lot
underway.
of that particular type of
Thanks,
fish
Nav.
on our first dive. In fact, all of the fish except for one was that were cutthroat eels. Also got some dead sponges in front of us. They typically turn brown when they die. They're either collecting sediment or I don't know, maybe
Why
the
don't we
tissue
swap at
turns brown when
3:15?
it dies as well. I'm not really sure.
3 o'clock's fine.
One of the
We should-
things it's worth mentioning here is that whenever you drop down into
Yeah. Why don't you introduce
into a
us?
location like this onto a ridge,
I
and
want
you
you to
can
do
get into a community like this, they often are different. Can have some of the same species as other communities, but then there's always something different. The ratio is different. And in this case, I don't think I've ever seen a community like this before, with these little small crinoids all over the seafloor, and then
You can get this shot if he wants it, but
and
don't
then
get
some of these
puppied
black
off too far to the
corals
south there.
the
Copy.
Stauropathes and maybe Trisopathes interspersed amongst them. So they
Watch
all.
the pilot.
Roger, pilot. Go
Sorry to
ahead.
interrupt you. Do you want a shot of this guy?
That's okay. Thanks. That's a fairly common one that we've
Cool.
seen before.
Keep moving.
Thank you. Now there's a fish right there. Maybe we could get a close-up of this. It looks different than the one we were looking at just a moment ago. He's at the bottom of the screen.
Okay, video. Let's see what we can do here.
Zooming.
So it's going to be in that same family. It's going to be a cutthroat
Take a
eel
bit
in
more
the
zoom.
family Synaptobranchidae, but I don't think it's the same individual.
Partial.
This guy has got some scarring on its head and on its body.
Little battle scars.
Right. And the things you look for to identify these eels are where the dorsal fin begins, and the dorsal fin on this animal is very far up, and very close to its pectoral fins. You can see it quite clearly. So this is definitely a different species than when we were seeing at that other site in the first dive.
Take a
Also,
bit
the
more zoom.
lateral line pores, the series of holes that you see along the side of the body, these tend to look white, and I believe this would tend to place this eel in the Iliophus group, but I'm going to go ahead and double check that.
Getting pretty close to a co-pilot. That's about all I'll do.
You know, for identifying
Yeah,
fish
okay.
and corals, the details really matter, and there was a person who emailed us this morning who was
Yeah.
talking about trying to describe some of the features that we need to pay attention to in some of these fishes. Okay.
Pull wide.
Take your video.
Nice follow.
Yeah. He was very dynamic. Okay, we got enough of him.
A little
Sorry.
sponge down there.
Yeah, that's one of these Hylonemas.
That ship move was 90, am I correct?
Yep.
Okay.
I'm lined up on it.
Done for.
Survey data is stopped at the reboot again.
Okay.
Last I checked, we
Seems
should
like
have about
these guys really like this area.
Yeah, this consolidated coral and ridges, so it can be pretty good for
yeah, we don't want to restart the computer yet at this
You know,
point because it's got
for a lot of animals.
the software
last
for the
year we came across quite a number of
fins
sites
on it as
that
well.
that were
We
ridge
don't want
sites.
to add
So
it
I
for
shouldn't
the
do that.
acquisition.
Pretty dense communities.
So
like
you can
this.
you can start that. Well, I was wondering,
So
you know,
again,
this
as I mentioned earlier,
what might
it
solve
may
the
be
problem is a
current
computer reboot, but
acceleration
I don't think we
over
want to
these
reboot
ridges.
that
That's
computer.
the reason why
It's got too many other things on it that are important.
we
I agree.
tend to find a fair number of animals on them.
So, yeah, we're
Trying to
we'll take a note of it in your
understand
log that
with
it's happened
these
a couple
dives. We're trying to
times,
understand what's going on down here.
and we'll have to
Got a beautiful little
see what we
purple
can do.
crinoid in the
Probably fix it tonight.
Cerametra group. Cerametra
They both have one.
Tricerealis, I think is the name. So got some
Pilots, that should be
ophiuroids
about
that
the end of the
we-
move there.
Okay. Did Sirius move
ophiuroids that we saw on some
sort
of
of
the
correlate?
one of the other
Doesn't seem like
sponges
we're over
as
the
well.
ridge yet, but
Just making
maybe
notes
we
in my
are coming
notebook
around
of
to it.
some of the different types of animals we're seeing at the
does it
site.
seem like
just so I
Sirius
can
is moving pretty well?
put some names in our dive summary report later on this evening.
Video, can we get a quick zoom on these
Not-
crinoids right here?
Of course.
It seemed like Sirius moved the same 40 meters that you called in. Yeah.
Not as much as I would have anticipated.
Okay.
I think partial I've got more.
Yeah. So there might be a little current
So
here.
what
Come in
it's-
just a little pretty quick.
It's clinging on to what appears to be a dead sponge stalk. And stalk also seems to have a lot of hydroids growing along
We're
those
just going to
things on
zoom
the
the,
left.
the lasers out of the frame.
So since they're
Unless
both
we wanna
hanging on the
could
same
use it.
side, they're probably just flags
Yeah,
on
come
a
on, those lasers.
flagpole.
Lasers out.
Full way.
It's a good point. I don't know if they kind of move around
They
depending
fell out of
on
my
the
light
water
pole.
current. I
Let's
assume
get
they
in there
do.
really quick.
Hmm?
So
I'll
does
come
this
closer
mean
to
the
the
current's
subject.
coming left or right because they're both?
That's my guess, but it was kind of more a question than
Yeah.
an observation.
Yeah.
Co-pilot, would
So
you
we're
like
gonna
to
go just
add another
bit.
15, 20 meters to that
I
move?
do see some little white specks floating.
We'll let
that
it settle
direction
for a
now and
second
then.
here. He's
Sure.
getting a long look here, and we've caught up. So
Okay, video, when you're clear. Let me
Roger.
go and pull out. Thank you. Okay,
So that guy next to
Back
him
in
is
your
doing
view,
the same
co-pilot.
thing.
Yeah, they're lined up identically. So Asako Matsumoto is asking, is there a strong current? I don't think it's strong, Asako, but I'll ask the pilot or the navigator. Do you have an idea of how strong the current is?
I do feel it in my face, so a east to west current. I don't have a magnitude yet.
But it's not terribly strong,
Not
is that right?
terribly strong yet, no.
And you're currently looking towards the northeast, roughly?
So Asako, he basically said, if you didn't catch that, that the current's moving east to west, and it's not particularly strong.
all right. Looks like Sirius is starting to settle more over the ridge here.
Another one of these Tritopleura sponges over there, and is that another fish in the water column?
Ooh,
Hid
he is
there, or
way back
just another
there.
one of those
So
hills?
D2's facing at 0.
Yeah,
6.
might be.
It seems to be
Yeah,
off in
again,
that
there's-
direction, so we could do 055 or something north.
I'd like to see something other
Northeasterly
than
from
the
here.
eels today. There's a lot of really interesting
do see
deep water fish.
I did see him, yeah.
Yeah.
There, you're right. That's
Let's try to
definitely the
sidle
same,
up on that and
yeah, the
see.
same
long
fam-
body type again.
Yeah, same
Yeah,
family.
maybe
again,
we stick to
probably.
060.
Video, come in, see if we can't get this fish for you so it's too
Roger.
far away. How much least do I got?
No, closer to 0.
Zoom quickly.
Like
Yeah,
065
so it's a
maybe.
different individual. You can just tell by
think
the
065 is
lack of
gonna-
scarring on
it's gonna
the
keep
head. That the other one
Ooching
had.
us a little bit more over top of that, but keep us
He
on track on
also,
moving along it at the same time.
He's got the white lateral line.
Although
but
he's
not
facing-
really
hold on.
cooperating at the moment.
Pilot.
He's swimming away.
Yeah.
Okay.
Anyway, definitely another
Come on,
snap to brankid.
Thanks.
So
Yeah,
that's
of course.
good.
Okay.
Yeah, that puts
co-pilot.
everything over there. I'm just trying to, sorry, pilot, we're just trying to sort out getting to the top of this ridge. High pack puts us there at this point. But still seems to be
There might be something
fairly
straight
east of
ahead
us.
at the top of the screen in the distance that could be a different coral colony. I wonder if we're going in that direction. Maybe we could take a look at that.
All right, let's go east a little further so we can
Yeah,
take a look
absolutely.
at that.
We'll actually we're headed that way.
Okay, thank you.
How about a 075?
Sure, that's fine.
You want to just do a 30-meter move?
Actually,
do-
let's stick to east or
Just do east
08.
another 10, 15 meters?
Yeah,
This could be a
at
dead-
least
mostly
not
dead
20.
sponge too. Hard to say from here.
Not 20.
Well,
Yeah, I
maybe
said at
up
least
there.
if not 20.
Some
20
of
might be
these,
okay.
White things that you're seeing on the
Copy
right
that.
side of the screen, these are dead sponge
Yeah, we
stalks.
can
They've
bridge a
actually
sniff.
lost their.
Yeah, there's something
the
else
heads.
coming out there.
I'd like to put another
Yeah,
ship
there's
move in.
another eel.
Is that an eel?
Yeah, same eel.
Do,
You've
uh,
seen
Oh,
his
that's
ro-
right.
you've
You're
seen
seeing him from
20
the back.
meters,
Okay.
bearing
Yeah.
090 at 0.2 knots.
Yeah, that looks like a dead sponge. One of these Tretopleura, mostly dead. That's to the left of the
Good
eel.
copy.
Thought it could be a different coral colony, but it's not. There's a nice anemone on the bottom over just in front of that dead sponge. And this eel is going to be different
this is the
than the
ridge.
last one we saw.
That's
But
the
a
hope.
different species.
Copy.
Tyros is all over the place.
You
I
can
don't know
clearly
what the
see that
currents
the
are like
dorsal
here, but
fin, that fin that runs along his back, doesn't extend nearly as far up toward the head.
Starts halfway down at least, if not further down, huh?
Right.
Just got his face into the current. And he's riding the current. He's getting close. And I'm gonna back off here. I think I'm gonna let him go. Bye. Okay. Come back down here.
so this, which way are you facing? Yes, okay, so that looks like the ridge crest
Yeah, I think.
right there. Okay, so you can sort of push off that way a little bit and try to get lined up with you on it.
Oh, lots of dead skeletons on the floor
Something
there.
like
These,
45,
I think, they
maybe
appear
40.
to be
Yeah, okay,
sponges
good.
on the right hand side, and on the left hand
that's
side,
good. We found
it's
the
not
top of the
really
ridge.
clear. I think they're mostly sponges.
Sweet.
Or
So
the
yeah,
remains
you've
of
got room
sponges,
to
if
push
you
up
will.
that way if you want it.
Copy that.
I don't know if they want to take a look
What
at this.
is that
possibly
thing that's hanging
sponge.
down there,
I think
dead
that's a
ahead?
separate.
Oh, it's
separate
a sponge.
sponge.
It's a stock sponge. It's a
Yeah.
different sponge. That's good. Maybe we could take a look at that.
Absolutely.
Yeah.
I have room to move.
Yeah, that's a good direction to push.
See Diva Amon, who's with the University of Hawaii, and she's actually going to be relocating, or has relocated already. She identified the eel as a synaptobranchid also.
That's a cool looking sponge. Very stocky. Stock. Got lasers, co-pilot.
Lasers on.
Okay.
That is sizable.
Yeah, so these are, we have seen at least something quite similar to this. It may be the same genus. And
Pilots,
but
now
not
that we're at
necessarily
the top
same
of the ridge,
species.
We
And the
evaluate
genus
the
is
bailout.
Caulophacus.
We keep
It's
it
in the family Roselidae. It is a
due
glass
west
sponge.
as ship has come astern.
And there's a bunch of these
rather come
glass
ahead now.
sponges in deep water that have got these stalks, and then the main body of the sponge is on the top
Yeah,
of the
we
stalk.
uh,
As you can see,
we should choose
that's a
based on the
good-sized
bottom,
sponge,
not really
as you
on
pointed
the
out.
requirement.
the
But
head on it alone. is
if
what,
we
about 30
stick to it being
centimeters
astern,
across,
then
maybe?
if something bad happens, then they just
yeah,
stop
trying to
holding
see
position,
how
and
the
will
laser
blow off.
dots compare. They're pretty much in the right place for that.
But
The other nice thing to see is
I
the,
imagine once we
this
get
whole
on this
rocky
crest
formation
that we
that
will
the
want to be
sponge is
going
sitting
opposite
on.
of.
Kind
of
of looks
it,
like
sort of
a,
this direction.
a layer
Kinda
sticking
south.
out. It's
Yeah.
likely a piece
Go
of
back to
sheet
rotate
flow
it,
or
please.
something like that.
Sorry.
Got your main HD.
So these
Okay,
are
video,
just
if we
thin
just zoom
lava
in, we'll
flows
start,
that
maybe
you
start
get
at the bottom,
underwater.
work way up, or
There's
Roger.
go
enough
to the top.
lava coming out at
You
the
want?
same time.
I'll hold it right here.
I'll hold the partial.
It looks like a little sea star at the base of that sponge. That's right in front of it. But yeah, looking at the corals all around here, these all appear to be primnoids from what I can see, and presumably they're very similar, and they all have got 2 branches. It's just
Oh.
very
Okay, we're
very
Free
odd.
to tilt.
Yeah,
This one here, right
tilt
to
up,
the
profile.
left of the sponge stalk itself, I
Tilt
think
up.
the polyps
Yeah.
are closed,
Come
and
out.
it looks like it's could be in the
That's
genus
cool.
Narella.
Ship
They
move
look like
complete.
they're closed up downward.
You could just tilt to the head.
Do we have anyone?
Scott
Yeah.
had a few
Sorry.
comments
Scott
on,
had a few
on,
comments.
Just so you know, you're
Pardon
falling
me, I have
off
to
and back
lean
a
over
little.
a little ways to look at the chat,
that.
and Jasper's monitoring that more actively. So it's
So
yeah,
can't
that
get
would be
both
great.
lasers
Thanks.
on it, maybe. Come on.
He's moved the screen around
There
a little
it
bit.
is.
So Scott France had noted that Narella alvini grows in the sparse 2-branch mode.
Okay.
Scott, would you
I wish
happen to
I knew
know
lasers
whether
off.
Narella alvini
Yeah,
has been
a couple of
documented in the
lasers.
Pacific? I don't know that species.
I'm gonna get a wide shot, and then maybe we can zoom on the
I
top.
think somebody said that was.
Sounds good. Yeah.
Thought I saw somebody say something about that in the chat as well.
It's like your sonar froze. I'm gonna get you
They were
restarted
asking,
here.
or they were
No,
saying
somebody
about it?
said they
Alright,
thought
ready
the
to
range.
tilt up.
Maybe it was
Yeah.
Scott.
Go back. Can you tilt me up just a little bit? Thanks.
Tilt up.
He
that's
noted
right,
that
sure.
the species is in the same depth range,
Just
but
want to zoom on the
the
top of the
question
sponge.
is whether or
Oh,
not
yeah.
it's in the geographic.
Okay,
Dark
yeah.
So
Yeah.
he's pointing out
Great.
that
And
Narella
tilt.
alvini is, as far as he knows, is only in the North Atlantic.
After the shot, you want to swap out?
Oh yes, I was looking at a different comment
Sure.
earlier
Sounds good.
for someone else.
Yeah.
You've got your camera back.
Copy that.
Okay, I'm gonna take a slightly.
What do we got
wide
left
shot.
in that ship move?
Ship move's completed.
All right, good.
Hey, video. You want to see anything else in this before we move on, or?
Nope, I think we're clear. Full wide.
Thank you.
Some of these primnoids that have more than 2 branches, they do look like they're Narella, the genus Narella
just-
to me at least.
Just push out.
But it's
now.
just not clear whether they're the
Um,
same species.
we're gonna Consider
just
settling
older.
up on the ridge now that we're here. I think so. See what you call. See what you think is sort of uphill. Let's line up on it. Up the ridge crest.
Yeah, there's a
Yeah,
different
okay,
type
so
of sponge.
045.
That's a bolosoma. That's
I'll
a
try to
different type of
match you
stock
there.
sponge that we're going to go over. We've seen a fair number of
045.
those on these dives.
We're on the ridge now. I don't really have much in Cirrus sonar. It's so flat.
I think
A few little blips here and there, but I think they're
I think we're
mostly
calling that
holidays
Species
I don't
3
think
at the
they're much of
moment, but I'm
anything.
going to double check.
or just noise. I can
Nope.
fuss with this a little and see
Species
if I can get
1.
the noise out of here.
This is
By
the ET
2.
sponge.
Go away,
Sometimes
noise.
it's pretty hard to remember the names of things when they're known as species 1 or species 3. Like, what number was that one?
Inventive names.
Right. There's a nice fan that's down there to the
Yeah,
right
okay.
of the sponge.
There it is. I can't tell if these spirals look like a bunch of
Amon
nothing.
also suggests that
This looks like the wall here.
Bolosomini.
I
It's
think I
the
agree.
stock
045.
sponges
So we
that
can
are
sort
in
of
the family Euplectellidae.
mosey in
They've
that
all been
direction.
placed in a subfamily called
Okay.
Bolosomini.
Are we to face the current?
That's it.
Or
Match.
swap?
Pilot, I wonder if we could get a look at that fan that's directly at the bottom of the screen, center bottom. This looks like something
Check out
that
the
we
fan.
haven't
How's that?
documented so far. Maybe I think it could still be a primnoid, but it could be something different.
We'll switch out a little bit.
So this span right here? I got
Yeah, that
this
one.
closer
That
to
one that you're
center.
closing in on.
Excellent. Get lasers coming back.
Lasers
Steve
on.
had mentioned that this could be Bolosomini species 3, and I would agree, I guess. Bolosomini, yeah, Bolosomini, I guess, is as far as we can go on that one.
Okay, but it didn't come in a little bit.
Partial.
Not too good of a background for this guy, but we can try coming in. Copilot, can you take my rotator? I'm trying to hold still here.
Yeah.
Thanks.
Tara
A little
Luke
up
is signing
here.
off. Good night, Tara.
Holding.
Glad you could join us for a while, at least today. We realize, of course, that it's pretty late where you are.
Max soon.
So let's see what the chat's going to come up with on this guy.
It's got someone sharing a ride.
Yeah, that's actually what's called an aplecophoran. It's a type of mollusk that we actually collected 2 of them last year and 1 of them so far this year. And there's a bunch of people out there that are quite interested in these mollusks. They're called aplecopherans because they don't have a shell like a lot of species of mollusks do. And Nolan Barrett also concurred it was an aplecophoran. And this coral is definitely a primnoid. And the question is whether it's also a different type of Narella. Just seeing if anyone wants to take a stab at it.
You gonna bring it back? Or I mean,
Those
it's
look
up
like,
down.
almost look like
you
they're
can tilt
stylasterids or something.
Unless you think we
Some
got
weird
it.
white things in the background.
Coming down. Oops, sorry, that was much quicker than I anticipated. I'll hold there.
Yeah, so I think my guess would be it's in the genus Narella. But, so Scott's not going to
Ready
go
for
any
full
further than Primnoa. It's
species?
probably wise, but
Yeah.
and I'm not as wise as Scott is, so I'll throw out a identification. It could be way off.
Can tilt down just a bump just to get the basin view.
Nav.
There we go. Nice.
Video's good.
Yeah. Go and pull out and roll. Thank you. All right. I think we're going to do a
That looks like a rather large dead
Yeah, it
sock or
it-
something like that.
it had a
You want me
good
to
long
put a
life.
ship move in, Copilot?
And, maybe it got too big for its britches, as they say, and toppled over. It looks like the base might have disconnected from the rock.
It just got too heavy.
Yeah, and there's a couple of them there. I think one's laying on top of another.
Yeah, looks
Scott
like
points out the law of
3
averages.
bases. Whole pile of them.
So we're having a little bit of a shift change here in the front row. And at some point, if they have a few minutes, perhaps they could introduce themselves.
Yeah. Yeah, well, actually, this is Nav. I was just going to see if this was a good time.
Yep.
Yeah.
Okay.
So up here in the front row, we have the couple members of the Global Foundation for Ocean Exploration as your pilot. I am Levi Unima. To my left is our chief navigator, Jeff Lanning, and to my right, who do we have? Nick Palenko as my co-pilot. And we got Roland on video. And do we have someone in the clipping chair right now?
Emily's
Emily.
in the
Emily is
clipping
back
chair.
there.
Yeah.
Excellent.
In the back, we already mentioned it, but the biologists and geologists are Chris Kelly and
Jasper Conter.
And we've got, even though it's really late on the East Coast, we've got a bunch of people.
'Kay, guys.
Steve Amon maybe in Trinidad,
Oh,
I
man,
think,
the
right
talkback
now.
on this is
Michael
zero.
Vecchione
Anyways.
is on. Nolan Barrett. I'm trying to remember where.
How you
Nolan
feeling? You
is-
getting a little
is
stretched
based
out?
of
You ready for a move?
right now, but Scott Francis is in Louisiana.
Go swap.
We've got a Sako Matsumoto, and she's not in Japan
haven't sat
right
here
now,
much
I
yet,
don't
so.
think, unless she returned. I think on the dive a couple days ago, she was off
I'm ready for a
Newfoundland.
move.
If I got it right. I kind of butchered
Let's
where
see.
she and
045.
Tina were. I got them mixed up, but it was
One of them was in the Netherlands, and the other one was in Newfoundland. That's what
maybe
I
a
remembered.
maybe a Sako's in the Netherlands and Tina was in
4-5 looks good.
Newfoundland.
Do you agree, co-pilot?
Also got
would
Allison
agree.
Miller on. And
That's
Andrea
exactly where I'm
Quattrini,
pointing.
Who's,
We're lined up good.
who's chatting in from the West Coast. So welcome to everybody.
30 or 40 meters, whatever you want.
210s. All right.
So you were just joining us. These are some sponges we saw earlier. The stock sponge is in the genus Caulophacus. And these kind of leafy-like
Bridge,
sponges
ROV
I believe
nav.
are in the genus Tretopleura.
We'd like a ship move, please.
So these are glass sponges. That
Let's
means
do
that they
30
have,
meters, bearing
microscopic skeletal parts
045
called
degrees
spicules that are
at
made
2/10
out of
of the knot.
silica. It tends to give them a very translucent appearance, particularly when you look at them under a microscope. And they're quite beautiful.
Good copy, Bridge.
And then some of these sort of brown ones, they're the same thing, but they've died, and so they tend to turn brown after they've died.
So compared to our previous dive, this looks a lot more busy
Thank you, Bridge.
with life here. We also have less pockets with sand in them than we saw in the last dive. But
Might be a good opportunity to zoom on the
Yeah.
Sirius cam. Maybe. Yeah, I don't know.
Yeah, it may be as simple as that, that the firmer, harder consolidated substrate attracts more animals,
Yeah.
at least this type of animal. Certainly there's plenty of animals living in sediment too.
yeah,
Have a cool shot.
So I think we might be waiting
Yeah.
for a ship move, if I'm not mistaken. Trying to see if there's anything here that we haven't imaged already, and I think we've got all of these animals already on the previous watch stand.
Yeah, watch lead. So we we landed a little bit west of the ridge, so we worked our way, and now we're
that's
on the
serious.
top of the ridge, and so we let things settle to make sure we stayed on top, and now we're moving up along the ridge.
Great, thank
What
you
was
very
that,
much.
Pilot?
So we will be able to
Pull
keep
out on Sirius.
pretty consistent moves
Once
up
we get
along
it
the
set.
ridge
Sure.
at this point.
Wow, excellent,
Make me
thank
feel
you.
a little better.
Yep.
Sure. Video, this is co-pilot. If we could go,
I definitely
zoom
like the
out
fact
on
that this
Sirius.
ridge is
Absolutely.
presenting a very hard,
Thank you.
solid surface here. That's a good sign.
better.
Sometimes
Cool
you
shot.
get these ridges that have a lot of broken talus all over them. I never
Maybe we'll
quite
take a look down
understand
here while we wait for the
how you
ship
can
to
get
move.
that much broken rock coming down slope on a very narrow ridge, but we've seen it. So maybe it's common, and maybe there's nothing unusual in it in your mind, Jasper. It just seems sort of odd to me.
yeah. I would say it depends on what kind of eruptions you've had before that built the structure of the volcano in the first place. These are, if you compare this to what we looked at on the first dive, that looked
Puppy
much
too
more
much
like
farther,
smaller
you have to
pillow
always tether
lavas
in his face, just so
coated
you
in
know.
the manganese.
Copy that.
And,
And
the pillow lavas tend to break up into smaller little pieces. These are larger structures. Some of these looked very much like lava flows and would probably take a lot more effort to completely break down to those kinds of smaller pieces we saw on the first dive. We're also, if you think about it, this ridge
Not too
has a
much
few
there,
ups
a
and
little bit.
downs, and so we're actually quite a few humps
Keep
out
hands off the
from
sticks
the
for a little
main,
bit, I can tell you how
main
much.
larger platform. So most of the sediment that might shed from the top of this guyot is probably not going to make it here. It's probably going to make its way downhill
Oh, you got
before
it
it
there.
you ever get down to this point.
So a lot of animals don't like that type of sediment coming down, and so we sometimes prefer to pick sites that are a little further away from
Let me
the
know when you
summit
got it.
for that reason.
Yeah,
I
we get it.
think we have a better chance of seeing some nice communities. So, Pilot, if you happen to see a possible candidate rock sample, and same thing for Jasper,
Let's do that.
came to mind because I see something on the bottom here that may or may not be suitable.
Yeah, we can look for rocks. Something loose. We're looking for something angular, probably. Or
Yeah.
Yeah,
Don't go
these
down
these
too far
look
on
pretty
that side of the
small.
ridge.
Can you turn the laser on?
Pilot, you're drifting over to the port side. In my view.
copy.
Do you want me to come around?
Yeah, the lasers are on. It's just a good
No.
distance away. You can't
Sorry.
quite see them.
So they may not be that small after all.
That's
We're
right.
just a little ways away.
Yeah.
So yeah, I just came off the ridge to see what was on this side, so I can't go down too far, but I can look up here would be in the realm of possibilities.
Yeah, the
Yeah,
little
just
pocket
that little
towards
pile.
the right
This
of the
pile.
where the lasers probably are.
There might be something there. That's right.
Yeah, a little bit to the right and above the lasers, maybe.
Put that one in there.
Yeah.
Think that's loose.
Yeah,
No
let's
idea.
go, Pilot.
Say again, co-pilot.
If you give the bridge an updated bailout,
Where do you have them at now?
when we were going west.
He's okay. He's along. Yeah, he's on the east side right now. So while he collects this, we'll leave it, and then when he gets back on top, we'll adjust.
Okay, video, you can come in just a little bit, see what kind of rocks these are here. Can't tell if they're stuck
Roger.
in place or.
Holding partial.
What do you think is that one right above the lasers? Would that work for you?
Yeah, that's the one I was looking at, a bit above where we are currently pointing at the lasers.
Pilot, do you think that's possibly loose, or do you think that's stuck?
I give it a 50-50. We can take a second and give it a poke.
Okay.
I have more zoom too.
Nav, what do you think about that? We'll look at it, watch the see what we got.
Sure. Thank you.
Ship's just about done moving. Sirius is
Video, you can zoom out.
comfortably far away from you. So, yeah, considering it's a pocket like that, I imagine these could be loose.
Okay.
That guy looks kind of. Does he look too big? No, he might not be.
He's on the larger side.
Be mindful of where you land, because you have to move the arm and you have all that stuff right there. So keep it so that you have somewhere to reach out without bumping any of that. There you go.
Couple biologicals close by here. He's a little bit below me.
Pilot, you mind if I turn my heading left a little bit and take a zoom?
Is the ship stopped?
Ship is currently stopped,
Yeah,
and
once if
Sirius
If Sirius is settled out, go ahead.
pretty close to settled out. You might make double sure that you're settled for a second here. But,
Sure.
We're going to be probably swapping around views to help him with
Copy that.
sampling.
Now, do you typically go into joy lock in these moments, or do you do a XY? Or?
I do a downward joy lock with an auto heading. So, but you want to get yourself pretty balanced already, and then go into joy lock rather than pushing the joy lock ahead of time.
Copy.
It doesn't take much down. Just 20 or 30% down is all you need.
I think that's pretty low.
Well, sorry,
New,
beyond. Yeah.
Yeah.
It's okay. Oh, you're afraid it's out of reach?
Yep. What do you think?
I thought you were just lined up on it. Yeah, no, that's plenty close.
Okay.
I think so. Yeah, you're all right.
I'll get settled in here real quick.
Okay. Your auto heading should keep you from spinning too far off, but if because the down it's spinning you, you can add a little extra left and that will fight that, and you'll have a bias here, but that's okay. A little bias between the set point and the actual is okay, just as long as what you like on the screen is good. So that looks okay to me. I think you could leave it alone.
Okay, Copilot, you can spool up hydraulics for me.
Copy that.
Yeah, just that back down to where you had it. You're swinging it. Those critters. There you go. Okay.
Pilot, I'm trying to enable hydraulics. Oh, there we go. Bring them up to about 600.
1500.
15.
RPM.
Hydraulics are at about 1500.
Keep an eye on that hydraulic reservoir percentage.
So,
It's at 64.
to prod a rock, you want to have closed jaws.
Okay. Yep.
You don't want to poke or pry at a rock with open jaws, or you can damage the arm. If you know after poking with closed jaws it's absolutely loose, then you may go with open jaws to grab it.
Okay.
Be slow and collected.
Copy.
And deliberate.
Okay. Hydraulics coming on.
For those of you who are not on the chat
What's
room
your grip
here,
force?
we're currently at 2,229
Feedback ratio
meters.
4.
Our temperature is
Co-pilot, how do you
1.9
feel about your
degrees
move? Are you
centigrade.
pretty settled there?
Our
Yeah, we're
dissolved
settled
oxygen
here
is
now
about
for a few minutes.
10.4,
All right.
I came
about half of what it
left
is
a
at
little
the
bit,
surface.
just keeps detailed view.
Salinity's about 34.6.
Okay.
We're on Beatty Seamount or Beatty Guyode, which is a flat top seamount inside.
Just remember,
the
just like before,
Wake
you have
Monument,
co-pilot to help
part of
you
the
with
Pacific
cameras,
Remote Islands
throw
Marine
anything
National
else up on
Monument.
that view, whatever you want.
Okay. I like minute cams are pretty good.
We are getting set up to collect a rock here.
Oh, he's way closer to the cameras.
Maybe your pilot cam would help you
Yeah.
tell where you are with
Co-pilot.
respect to that.
Can you take my pilot cam, Ford PTZ,
This one.
this guy, and then turn him to see the arm? Yeah.
Right there.
Yeah, get the arm in view there.
There you go.
That biological is rather close to me. You don't want to hurt him.
So now if you push down, you're going to push yourself off. If you just pry a little on one lip or the other, more likely to stay on the bottom.
So for those of you that are looking, we are trying to see if we can
Underneath that lip.
collect
and
a
then
rock in
try to
this little
lift up.
pocket here. We've so
Yeah,
far seen a lot
You're
of
just trying to
very
wiggle it a
solid
little,
rock
make sure that
with,
it's loose.
thick manganese covering, and we're just trying to take a look here to see if this happens to be a loose rock, of which we haven't seen a whole lot yet.
Looks loose.
It's a pretty
It
big
does
rock,
look
right?
loose.
It is a pretty big rock.
It's doable.
Well, it's like,
Not even. I think it's about
Let's
this
go.
big.
well,
I think that looks similar in size
Oh,
to
he's
the
moving a
rock
little
we
bit.
had
Yep.
But
the
yes.
first dive on the first rock.
So it's roughly, what, 40 centimeters wide?
I think it's about 35, 40.
I think it's too big.
I can't see the depth dimension. I've no
Try to
idea
do
how-
30 or less, but
Well, let's go ahead and grab that guy and get on, since we have a little bit of a shorter dive, and if it's not too much of a problem being slightly over your size. Looks like it's
be very
loose.
careful of the coral cutters.
Yeah, watch lead. I was talking to my co-pilots here, and it is pretty big, so we're going to try to grab it. So hopefully it's not too big. But
Not knowing the depth, we're going to see how it goes.
yeah, we'll see what we can do.
Okay.
What grip force do you have?
8. Can you bring my co-pilot? Can you bring my pilot cam up to the subject?
Sure.
Yeah.
So for those of you tuning in from home, the red dots are the laser dots. They're 10 centimeters apart. We use that as a scale, so we have some idea of the size of the things we're looking at. And that looks like a solid piece of manganese.
Yeah, could be.
We're going to go port or starboard. Starboard box co-pilot.
Starboard box.
So you can bring that in.
Starboard wing inboard.
Yep.
Jasper, what were you referring to by manganese?
So,
if that isn't a
perch.
setup question,
Looks
I don't
like
know what
you're
it
okay.
is.
Gee, that's a
But,
difficult one.
Try to move real slow
So
and
most of
gentle
these
with
rocks
the arm.
at this depth for this
You can
age
swing yourself
have
around
a
lot.
layer of
Are we going
manganese crust on
to image
top.
this one, or
And
Yep.
certainly some of these older
so it's up
ones,
for the video, and
the
then
manganese will effectively
spin around.
break off,
copy that?
and you'll end up with just
Trying to bring the
slabs that are
starboard
basically
wing
just a crust. And there may not be any rock inside of this at all.
There we go.
The platy look to
Okay, I'm gonna
this,
spin it a little bit here.
I'm not so sure that there's much else involved
Show
in
good,
manganese here.
clean, safe coral cutters.
It could be something. That's
Yeah.
a flat side there.
It has a bit of
Okay,
up and
keep
down
spinning.
depth. We'll have to wait and see what we
Now to
see and what we
continue.
see at the
Continuous
surface.
mode. Click the square button.
Although I did see a little
And we're
Okay,
up and
keep
down
spinning.
depth. We'll have to wait and see what we
Now to
see and what we see at the
continuous
surface.
mode. Click the square button.
Although I did see a little
And very careful.
You have
yellow
to index in
scratch
to be able to do it.
on the other side where
Index
the
in and then-
the grabber was. So
The side, though, you don't see it, so maybe
there on the end,
yeah.
this is probably sediment.
Right.
Is that an okay sample?
Well, we have it already, so
Okay.
you're close enough. You might as well put it in the box.
The one in
Just
the box.
oftentimes you just can't tell until you
No.
actually grab it. It's really hard on these really old seamounts where you can expect substantial amount of
Yeah.
this manganese crust on the rocks. Tricky part's finding a rock that's got some basalt in
A
the
rock
center,
that's
yeah.
actually volcanic. Yeah, that's the hardest part.
Yeah.
And so
So there are geologists
So,
out there
remember
that
when you're
study
putting
the
stuff in
manganese
here?
crust too, so
The-
it's going to be
To
valuable
not push
no matter
down
what.
too hard.
and the sediments.
It's all the way inboard. It's not
It's
supported.
all the way
depending on the
inboard. It's
kind of
not supported
sediment,
by the wing anymore.
may be interesting still for different
That's how big she is.
different types of research. Sometimes the sediments contain
Fit in
some
there, okay.
volcanic minerals that we can separate out, and then we can still get some idea, at least of the chemistry.
Index
Or
real quick.
sometimes the sediments are mostly material from upslope from the reef that sits up top the Guyot, and In that case, you can look at microfossils to get some idea of the time that the reef was deposited. So there's still things that can be done with this,
Yeah.
even if it's not a volcanic rock.
So you about ready to set this up and note the collection on the collecting?
Yeah, let's do that.
Okay, I think I'm gonna drop it from here.
Okay.
This is going in starboard.
Starboard rock box.
Try to
Yeah.
open your jaws slow. Yeah.
Okay. Packages away.
I think it was starboard. Yeah, it is.
Yeah, looks like it.
It's in the bag. So what's happening and what Jasper's doing right now is he's taking what we call an SCS snapshot.
Pilot, like
And
me to
what
bring
that
in
means
starboard
is
wing.
as soon
Bring
as
it
the
outboard.
collection is
outboard
made
and,
and it goes into the box, he's
Disable
got a
HPU.
form in front of him on the monitor in front of him, and he clicks collect, and he punches in that this is our first rock specimen 1 GO, and where it went in, and a tentative ID, and then he pushes a button, and that automatically grabs the latitude and longitude, the depth, dissolved oxygen, temperature, salinity, and all this other ancillary data at the very moment that the specimen is collected. And then later on,
Okay, pilot,
after
you want me
the
to-
Still got hydraulics
dive is
spun up?
I do.
completed,
Why don't you ooch the drawer?
we
in
will
while you're at it?
take
Both wings
this
out. Ooch drawer.
set of data. So if we make multiple
Bring the drawer back
collections,
inboard.
Yeah.
we'll get
Or
this
bring the
as a text file at the end of the day, and as
Retract
the ROV is
drawer.
coming up to the surface,
Retract.
Bottom
we'll
right. Slider. Drawer.
put
Retract.
that information into a format, a template, a label format, and then we'll print
Okay.
out the
Push the
labels.
port wing outboard.
That'll go in with the samples and have that all ready to go by the time the rock or the biological specimen reaches the
Nice. Okay.
surface.
We're in.
So
Thanks a lot.
it's
Okay.
really nice. The label is very, very
HBU.
complete, and everything's
Ramp down.
automated. So it's a
Yep.
very easy thing to do.
Do I disable it or just right?
Now, what will happen to that rock on the
Okay,
once
so
it gets
I'm
on
gonna
the ship?
pick up, and I need you to come up with me.
We'll be given
Copy
the
that.
all clear signal once the ROV is secured on the deck and they've opened up the collecting boxes. We'll get the all clear, and we'll go down with buckets and hard hats, and we will grab those specimens and carry them back into the ship lab. And there, first thing that'll happen is we'll rinse that rock off, and then we will put its label on it, and we will photograph the rock, and then we'll look for commensal animals that might be attached to it, and if there are none, then we'll set the rock in a tray to dry for several days.
Yes, touch more. There you go. Next spot.
Nice. And previous setting was I think 045.
Once
It's
the
working
rock
pretty
is
well.
dried, then we will
Sweet.
Getting pulled a little
go
bit.
ahead and bag that rock
Both?
with its tag,
heading.
and we'll place it into a rock holding area.
Here I come.
HiPAC has the ridge at like 46.
Yeah.
Saw another little type of sponge there, probably a small polyopogon.
Would you like it?
Another
Like a snap on the
hexactinellid.
polyopogon, or
We've got a pretty decent community down here. Not tons of diversity, but we've got a fair number of animals on the bottom.
you let me know if you see anything you want to catch. Otherwise, I'm gonna zoom along here.
Okay, yeah, maybe what we ought to do is get upslope a little bit.
Okie dokes.
He's got you at 52. When you guys are ready for a move.
Yeah, we're gonna get lined back up a little bit here.
Okay.
once we got a little off track.
Some more dead stalks of sponge. These are these white things that are sticking up. They're probably something like Caulophacus that lost their heads. We got a sea star right there in front of us, a Brazimjid.
Is this one typically your starboard rail cam?
Yeah. I put it on that for sampling. You can move it back to starboard rail if you want.
So a fair number of dead sponges. We see
9,
them
I
every
think.
once in a while, but often not quite as many as this. So it's not really clear. Maybe they just died over time, but they haven't fallen off yet.
That's right, they were all laying over on the previous dive.
We've had some laying over here, but we've also had a fair number that are still stuck to the rocks. So maybe there isn't really strong current here. I'm not really sure what that means. There's another one of the Bolosomini sponges that Diva had helped us with the identification on earlier, that stalked one. And we're still getting plenty of these little primnoid corals all over the bottom.
Okay, ready for a
as
move?
well. These little two-branch
Yeah, I think you're stretched out ahead of me, pilot.
that.
octocorals.
Yeah, let's do
Pulling
it.
me a little bit.
Yeah.
45, still happy?
Um,
Closer to 40?
yeah, maybe ever so slightly closer to 40. I'm actually lined up uphill at 35-ish, so you can split the difference maybe.
35 looks good.
Yeah.
Okay. Cool. Bridge over nav. Ready for another ship move. 30 meters, bearing 035 degrees, 0.2 knots.
All
That
right,
guy
we're
up
getting
there,
the ship
that
up and
sponge
rolling
is-
after that sample.
Yeah.
So
Like
if there's
a
something
hand.
in the immediate
Yeah.
view that
That's
you want to
correct.
take a look
Good copy,
at,
Bridge.
we've got a couple seconds here.
How about the anemone? Because that's a little different. I think
Okay.
we've got close-ups on most everything else.
All right.
He appeared right on cue.
Yeah, that was totally by coincidence.
Right.
About 10 centimeters,
Move initiated. Good copy.
It's
Thank
a
you, Bridge.
reasonable size anemone.
and we can do lasers off, and we got a little tug from the tether, but we can come in. Partial.
Roger.
Thanks.
Want me to come down a little bit, pilot?
Nah, you can leave it. But you can get the lasers for us if you don't mind.
Been partial.
Lasers.
So I think this anemone is going to be in the family Exosalactidae.
Nick, you can also,
I guess from
Press
this
and hold
view.
the lasers button that's illuminated.
But,
Press and hold that.
let's see if some of
Yeah.
the others will weigh in on what this could be.
There, perfect.
Little shortcut. Great.
Anemones, we don't have very many anemone specialists here on the chat right now, so we're kind of all at the same level of guessing as to what these things are that we're looking at.
Once I get settled in, get a little tug.
Almost looks like an old coin.
Yeah.
Sorry for the bouncing. He's just out of view. I'm just at the end of my leash here. But
Well, even with that, we get a pretty sure it's in the family Exosalactidae. It was a good to zoom it.
great. Okay. You want to come out wide? Video?
Sure.
Thanks.
Full wide.
Back up a little bit more, just so that we're not bouncing around with all our shots.
And Nolan Barrett also thought it looks like it could be Exosalactid as well. He's looking at the OER animal guide too, so you don't know about this guide. You should, if you're interested in deep sea animals. You because this guide has over 1,700 images of deep sea animals that were taken last year by the Okeanos Explorer and the D2 ROV. And they are beautiful, beautiful images, and we consulted taxonomists around the world to try to get the best IDs we could for these images. So you deep sea animal people out there, you need to go on the OER website and you need to find that guide, and you will no doubt start to be very proficient at guessing like we do what these animals are that we're looking at.
Yeah, ship is about halfway through the move. Yeah. A 30-meter move.
So we're looking around over here a little bit more because we're waiting for the ship to move.
Yeah, so...
And then we can move on up a little more after that.
So what's ahead on this dive? We're kind of on a narrow part of this ridge right now, and then as we move up slope, we'll come up over a little bit of a peak on the ridge, and then it'll kind of flatten out. And it's kind of interesting to see whether or not the community changes when you get sort of a topographic change like that.
Yeah, I think it's going to mostly depend on whether or not there's going to be a lot of sediment on top there, or if it's going to be kind of similar to this.
Right, or there could even be some type of nodule beds up there on the flats as well. We just don't know. Some of these little brown corals in front of us, these are black corals. We saw these earlier, the Trisopathes, it looks like. That's what we're calling them. They could also be some type of a branch
come down a little bit.
and
maybe 3 or 4 meters. If you don't mind.
Sure.
Thanks. Just give me a little.
Could even be a Bathypathes going off screen on the right.
Do you want to tack on another meter or another move?
I'm happy to keep them coming. Yeah. I don't know how close we are to the end of the last one, but I think we should keep them going. We want to make some headway.
Looks like a little flat. Maybe we'll do a 50-meter move this next one.
Yeah, there's nothing imposing on
Looks
your
like
sonar.
ship is almost in position. I can tack on another 30.
Yeah, let's keep them coming.
Copy that. Bridge over nav. Can we add another 30 meters to that last move at a bearing of 035? That is correct. Good copy, bridge. Good copy, bridge. Move initiated.
So this dead sponge coming into view right here, that's a different type of sponge, but we haven't seen a live one yet. I've seen a couple of dead ones there, and I'm hoping we'll see a live one of those at some point, and then we can kind of mark that as being at this site. We don't typically count the dead ones as...
Oh, they don't count towards your...
Is that thing in the background there something that might be similar but alive?
Yeah, but that's like the one right on the left here. I was talking about the one that's sort of center now. This is something that's called Aspidoscopula is my best guess on it. But I've seen several of these dead skeletons, and I haven't seen a live one yet. But there probably will be a live one, I think.
We hope.
Yeah, we hope. I want to mark it, but I'm not going to do it. I'm going to resist. Yeah, and then the one in the background there is one of these Tritopleuras again, just like the one on the right. They used to call these leaf sponges because they look like leaves, upright leaves. Yeah. And you just do a little nudge on them and they fall right over. They're pretty...
We won't do that then.
No, you don't do that. And I know this because we accidentally bumped one year in the submersible, and I couldn't believe it. Yeah, it was oops. And felt a bit bad about it, so it became a specimen.
Ah, yes. Might as well take it home at that point.
It got bumped over for a good cause then.
Yeah, bumped for a good cause.
Yeah, for geology's sake, we'll take a quick. I know, just because we're waiting for the ship, there's a bit of something here. I don't know if you have any interest in looking. I just saw it on another camera. I thought maybe you'd want to see. We don't have to zoom if you aren't intrigued by it.
If we're waiting, let's see what this stuff is. This is
a little pocket of sand, it looks like.
Well,
it's sort of a mix of...
Why don't we take a quick snap?
Snapping.
Yeah.
Worst case scenario, it's boring and we move on.
It could be these barnacle shells is what I'm sort of wondering. I think it could be.
Holding partial.
Okay.
Definitely animal shells of some kind.
Oh, okay.
Mixed with little chips of manganese crust.
Yeah. I think sometimes people call this stuff, they speculate it could be pteropods shells, I think, but I don't know those animals very well. But again, these could be barnacles as well. Barnacles are made up of a series of scales, and when they die, the scales kind of come apart, so it doesn't just look like a typical dead barnacle. You see these pieces all around.
All right. Well, we don't have to keep. Yeah, we can come out, I suppose.
So are these
maybe we would be
barnacles
curious.
that would be around here, or
Go on.
I don't see any. I haven't heard you say anything. I don't see anything that I would think is a barnacle.
No, haven't seen things. Again, these may have sort of slid down slope or something. Well, these little stringers here up on the left, these are probably live barnacles here. Maybe we could take a snap zoom and confirm that this might be the source of these shells right here.
Oh, yeah, okay.
Yeah.
I'll get a little closer so we can get a good zoom on it, and we'll see if we can determine whether they are bivalves or some other thing.
Yeah, that's a good point. That could be the other thing that they are is bivalves, but
Okay, video, you can start in.
I think these are going to be barnacles.
Not sure.
Thanks.
Yeah, I think so. There's a little anemone right there in front, and a little squat lobster crab.
Come in a little further. Oh yeah, they are little barnacle-looking
That's
things.
nice.
Yeah,
That's max zoom?
Yep, it is.
Thanks.
they're white, but they kind of look dead. I mean, they're hollow.
They're hollow,
Yeah, they do
yeah.
look hollow. Kind of scanning, looking for one to be not so empty.
Yeah,
I don't see one.
I don't either.
Well, I have a little leash so we can run off whenever you're ready.
I think that's fine. Thank you very much.
Yep.
That would be great. I like the way you call that leash. So, and what he's referring to is that this vehicle that you're looking at, the HD or the camera 1 view right now. This is the actual main ROV, the Deep Discoverer, and it's tethered to the camera sled, the Sirius ROV, which is above it. And if you're looking at camera 2, that particular view is from the Sirius vehicle. And if again you haven't done this, it's sometimes worth toggling between the two because they're very different views. The Sirius really gives you a sense of a larger scale of the actual feature that we're on, and you can see the ROV moving across the bottom there. And then of course camera 1 gives you the beautiful imagery from the main camera.
Do you see that pink? Is that any? Do we know what that is already? Any interest?
3, 2, 1. All right, we'll keep going.
We definitely have entered a field of stocks.
A field of stock. A stalker field? you
Oh
mean?
boy. I think
Oh,
this
yes.
conversation is stopping right
Right.
there.
Reminds me of truffula trees.
There's a little Fareed sponge. I'll make a note of that. That's the first one that we've seen.
This guy here.
Yes, that's right. It's Furia near Oka recta. Very small guy. And that used to be a live sponge, and it's pretty well dead, although those little white parts on it are probably still living parts of the same colony. Most of it's dead. It got pretty big size before it died.
But it looks like ships in
Now
position.
this is
Okay,
just
tack on
rude,
another
the
one.
sponge right here to the right. So what's happened here is that
Hmm?
Let me tack on another
the
move.
white part of the sponge
Good.
I
Yeah,
think
sure.
is
Ship
a
stopped,
different
so,
sponge
okay.
than the brown part.
Yeah,
the
we
white
got room.
part did not respect the fact that the brown one had died. It landed on the brown one and now growing
Started
off the
growing.
brown. And
It'll eat the dust soon enough. the
Bridge
rest of it.
RV nav.
yes, because
Ready for another ship move, same as last. 30 meters, bearing 035 degrees at a speed of 0.2 knots.
Sorry, is there something you're interested in, or you're just noting? Keep rolling.
Yeah, we're babbling on.
Okay.
about
Good copy,
things.
Bridge.
Well, we're trying to learn from what you're saying.
That may or may not be a good idea. So there's a little coral right in the center that's a little wiry curved coral.
Yep.
I think it's the first time we've seen, I think it's the first time we've seen the species. It'd be good to do a zoom on that.
You
Good
wanna
copy,
zoom
Bridge.
on that?
Move
All right.
initiated.
Yeah. It's true, it's not the most glorious thing out there, but it's different.
Well, science isn't always glorious.
Yeah, and I grew up with the Charlie Brown Christmas, and so the little sort of runt little tree there deserves some love too, right?
That's
Of
right.
course.
It's got 3 needles still, but it'll do.
Yes.
Okay, video. Can start in on it. We'll make it as glorious as we can here.
Absolutely. We're in the business of glorious. Holding partial.
Okay.
Okay, so this is what I thought it was. I haven't changed my mind, let's put it that way. It doesn't
Going a
necessarily
little
mean
further?
it's...
But you're not going to tell us what you thought it was.
Yeah, I'm going to see what the chat comes up with.
And then say that you were right.
Yeah, whether
Yeah.
I was right or not, I can ask it. I know Scott's there, and he was only
a couple
willing to
more
go to
seconds.
Primnoid
Roger.
last time, on one of these other things, but I want to see if he's
Continue
willing to go
on.
a little further on
'Kay.
this one, and
Clear.
Asako
Thanks.
too.
Thank you.
They both just go Coral Primnoid Whip. Oh, man, you guys, you gotta just walk out to the edge of the plank there sometimes. Get a better view of the water that way, right?
Get a little pushed
There you
out.
go. Scott did. He fired off a genus Candidella, which I would agree with as well. It could be something we call Candidella gigantea. The confusing part is that it has whorls of 4 polyps, that particular species. From the type specimen off of Hawaii, it has only 3 polyps coming off of it. So, but all other aspects of that look quite similar to that animal. Why do they call this little runt of a coral gigantea? And the reason being that the polyps are really large.
Is this a live version of what you're looking for coming up, center, in a way back?
It might be. Yeah, it could be. I think you're right, Pilot. I think this is. It's a smaller one, but
Smaller, but it's
Still
there.
counts.
it's there. It still counts.
Because it's not brown yet.
Yeah. I don't note the sizes in here in my little
Oh,
cheat
okay.
sheet
There you
for them.
go.
Yes, it is.
All right, I
Good
can't
try.
really land because I have lots of dead sponge. I guess it's not a big deal if it gets brushed a little.
And that one has a friend as well, it looks like.
Yeah, I think that's another one. Friend or brother, you know.
Brothers in arms.
Yeah.
Yeah.
Well, it literally could be a brother. I mean, some of the theories about why you can get a high number of the same thing in these areas could be that you have a pioneer colony or a bunch of colonies, and they kind of just spread out from there. We also have a black coral
All right,
over
video.
to the
I'm gonna settle in
left.
here, and then we can zoom away.
Copy that.
All right, have at it.
Just go for a zoom
Not only
first.
do we get treated to a beautiful close-up of a glass sponge, but we also get a crinoid that's...
Are we in luck? Is this the one you're looking for?
Yeah,
Yes, it
270 is good.
Thank you very much. This is
Awesome.
something called a Spidoscopulia.
Thank you, Rich.
And
Okay, I'll take
it's
it
in the
first zoom.
family Phariidae. And the crinoid, well, we're not going to go so far on the crinoids. We're going to go to maybe suborder or something on this thing.
Sorry, video.
No worries.
And before we leave here to the left, Sako Matsumoto had pointed out that there's an Antipitherian.
All right, come wide real quick.
Okay,
Let's try to
so
grab
we have time
the-
for a quick establishing.
quick. Yep.
It's, let's
running
see, a
low
little
on
more
time.
to the left, I think.
Yeah, we're doing a quick, just for the archives, quick establish
Okay,
shot.
that's good.
Sure.
And then we'll
Okay.
move over to there.
That's him, the
Yep,
little
I
orange.
see him. Looks sort of bathy like, but small.
Yep,
And
that's the guy.
Without the long stem.
Ship is almost in position.
Oh, good. Okay. We don't have
Oh,
a
and he's
ton of
hiding
time, but we
behind someone
will.
else.
Well,
We'll snap.
both of them
Get in
together
here
are
and
two
snap
different
in.
types
Yeah.
of black corals.
they're
It'd be good to get them both in the same frame
We will
because
here.
Scott is here now.
Come on
and
in.
I'm not sure he saw what we're calling
A little wobbly,
Chrysopathes
but we'll
over
get
here.
there.
That's all right. Just gonna frame them. Go in a bit further.
Goodness.
There's the one, and this in the background is the one they wanted, right?
Well, both of them actually.
Oh, okay.
Because we've got
You can rock focus
Roger.
as you need. Or rack focus, sorry.
So Scott has confirmed they're both black corals.
Oh.
And
Anything you need to see in particular? We don't have a ton of time right here.
no, he's confirmed the one in the back is Bathypathes as you suggested, pilot.
Okay.
So that's good. And then I think he's gone Trisso or Hexapathes as the other one, so that's good as well.
All right. I gotta
Come
get
on.
up, and I'm gonna give
Clear.
a good push out because we're about to do a watch change, and I want to give him a little time to jump in the seat here. So Josh will take over in a couple seconds. Make sure you're coming up. Oh yeah, you are good.
So while they're doing this watch change, I'll just remind everybody what's going on here today. Presumably you know this is the NOAA Office of Ocean Exploration and Research that's bringing you this live broadcast from the NOAA ship Okeanos Explorer. If you're watching it, you had to go on their site, so I assume that you know that already, or you're in one of the exploration command centers. But you are looking at a live feed from the ROV Deep Discoverer, We are currently on Beatty Seamount, which is a guyot, or a flat-topped seamount. We think it's very old, presumably Cretaceous.
Yep.
And our current depth is somewhere, let's see. Our current depth is about 2,200 meters or so, 2,225 meters. And we're on a bit of a ridge feature that's coming off of the southwest side of the seamount. And we're moving up the ridge. And we've found ourselves into a fairly dense community here, but a lot of really small stuff. No huge bamboo corals yet. In fact, we haven't documented a single bamboo coral yet on this dive, which is pretty unusual.
What we also have seen is a lot of these sort of layered rocks, all coated in this black material that we call manganese crust. We picked up one rock sample once we found one that was loose, and it looked like it had a sort of yellow-orangey color inside where the grabber arm grabbed it. So it looked like it was a sedimentary rock, potentially some what people refer to as hyaloclastite, which would be volcanic little pieces, shards. In which case, these layers here might just be sort of sedimentary layers that got covered in manganese.
Video, can you make sure Sirius HD is all the way wide?
Could you repeat
So
that, Cobo?
Could you make sure Sirius HD is full wide?
Yep, it is.
Thank you.
Here.
It's a little close.
Change in the community so far. The little leggy things on the bottom we've identified as small primnoid octocorals, and these large sort of leafy looking sponges. There's a couple of them. One of them's called Tritopleura and the other is Spidoscopulia. These are glass sponges. There's several different types of black corals. There's Bathypathes and Trissopathes or Heteropathes. And then like this dead stalk, so there was some other types of
pardon
sponges.
the interruption. Your conference contains less than 3 participants at this time. If you would like to continue, press star 1 now, or the conference will be terminated.
So we're seeing a nice little bunch of black corals here, a little bit higher density, and really nice boulder feature up ahead. That sponge on the boulder, we've seen a bunch of those today. It's in the genus Bolosomini, or in the subfamily Bolosomini, stalked euplectellids.
Thanks.
And now there's something white up ahead in the background that looks like it could be the different coral colony. I wonder if we could take a look at that. In fact, we're finally seeing something different.
Heading that way.
Or it's a really good-sized Tritopleura sponge. Now that I look at it, it may still be the same thing, but a really older colony. Guess I keep hoping for some bamboo corals, but no, it's the same sponge. So we probably don't need to zoom it. I was mistaken. So
Just a quick, and then
I'll confirm it.
Yep.
It was pretty close, right? I said it was a coral and it was a sponge. That was pretty close, don't you think? Yes. It's wishful thinking, you know. You just there's got to be some glorious big coral fans on this ridge somewhere. Yes. Yeah, maybe the little vase that's coming into the center of the view, it's off to the right slightly. That could be a different sponge. Oh, even this bigger one here, even right on the edge of the right. Yeah, that's the first time we've seen one of these, so maybe we could take a peek.
Just come on in video.
Okay.
Holding partial.
So I think this is in the genus Dictyalis. It's I think it's a euplectellid. I think that's the family.
I'll take a bit more zoom on the top.
So this is indeed a vase sponge. On the top of it, it has a big osculum or opening at the top that's covered by some fused spicules or skeletal parts, and that's called the sieve plate. Now we're looking down at the body of the sponge, and one of the characters of Dictyolis is a lot of these side or lateral oscula tend to be in sort of horizontal rows, which we're sort of seeing here in contrast to some of the other vase sponges. So I'm pretty sure that that's what this is.
Establishing.
And for those of you, again, who haven't heard my spiel on other dives,
go ahead, Bridge.
We believe that the water comes into the side of the sponge and then collects in the center and then goes out the top of the sponge. That's the way these things work. And as the water's passing through the wall of the sponge has got special type of cells that can extract
Copy that, just keep me posted.
very small animals and perhaps particulate matter from the water.
What's that, Dave?
Nothing. You were
They're filtering the water.
hitting a sponge. It's all right. It's not a danger.
Come on in video.
Partial zoom.
This guy, it's probably the same thing, and he's got a crinoid on top. Again, one of these comatulids, but this angle, looks a little bit
Go ahead
different.
and get to crinoid.
So these little side holes are not quite so horizontal. Yeah, I'm going to just say it's the same thing because there's one nearby as well, even though I'm not absolutely positive. But definitely a glass sponge, definitely I think in the family Euplectellidae.
Does he give you at least that warm, fuzzy feeling?
Yeah, I mean, sometimes it's hard to tell what they are,
Clear.
further
Coming
than that. The whole sponge taxonomy is based on their microscopic skeletal parts. We're trying to get better and better at identifying the species from imagery. It can be challenging sometimes.
Okay, pilot, you're starting to get out ahead of me. If we'd like to get moving, we could probably do that.
We're not moving.
I believe we're stationed. Talk to him in a minute.
You okay with that, pilot?
So Patty Fryer.
Okay, we're gonna move. We're not moving the ship right now. He just wants to turn.
At the University of Waimanoa, she was on the last
Bridge,
leg.
that's
For those of
fine.
you who are watching the last Marianas cruise, she was noting that the rows, the holes in the sponges were not in horizontal rows, and I tend to agree with you, Patty. I was just a little bit reluctant to create a new animal there, just from that kind of overhead angular view. But there's a very good chance it was something different.
And Nav, we've settled out here, right? Should be stationary.
Yeah, ship stopped moving a while back. You should be stationary. If you're not seeing anything, then you're not going to.
Yeah, I think I'm stationary.
You should be. The last move was 353, and it was only a 30 meter.
Oh, okay.
So we're slowly approaching a little high and I'm wondering whether we're really going to see much of a change in the terrain. I think it could be pretty just a little bit flatter, consolidated rock here. And we're looking at an anemone. This is Exosalactus, like the one we imaged a little while ago. These big deep water anemones. It's one looks to be almost the same size too. The other one was about 10 centimeters across at the disc.
We lose the skids. Thank you.
Shot gives you an idea of the density, even though there's a lot of small stuff. There's quite a lot of stuff per square meter growing on the sides of these rocks. It's just, only 20, 30, 40 centimeters high. And it's all very sort of leggy type of colonies. There's one of the Narella fans down there. Lots of Trisopathes or Heteropathes, these brown ones here. Yeah, that's a
thicker white one.
Yeah, that's a dead thicker white one. That could be a dead isidid, actually.
Bridge, Harvey Nav. Hey, what's your current bailout plan that you have listed?
Named right. I can do that one.
Copy that. We're gonna keep it right there. Is
And
that ship turn all complete?
Right.
We have a number of coral specialists on the chat participating today. Asako Matsumoto from the Chiba Institute in Japan, and Scott Franz from University of Louisiana, Lafayette. And I don't know if either one of them has seen this sort of density of these possible heteropathes, Trisopathes, these black corals. These are these brownish looking things that we zoomed in on a few minutes ago. But it seems like a lot of them. I don't remember being on a dive where there are so many of them concentrated like they are here.
Copy. Thank you.
Now, one of those boulders would be nice to take up.
Yeah, you copy that. Our last one was around 35 degrees. Looks like that's still uphill on Sirius. Are you guys okay with another one at that?
Yeah, I like 35.
30 meters, 0.2 knots, good?
Sure. Perfect.
Okay. Bridge, Harvey Nav.
And there's a different
Let's go ahead and put our ship move in. 30 meters, 035 degrees,
right
0.2 knots.
That we have.
You mean to the left bottom?
Yeah, that one that's sort of, it's got a little bit of a column
Good
coming
copy.
up and then sort of a bowl, a thicker one. It's some type of a glass sponge in the family Pheronematidae and perhaps even in the genus Poliopogon, but it's exactly what species it is, I'm not sure at all. It's got a bit of an osculum on the top of it.
Nick, come on in video.
No, I guess it doesn't actually. It looked like it had a little bit of a square area that was going to turn out to be an opening, but it doesn't. It's closed. You see to draw this shape because I'm not really sure what it is. One of these days we're going to go ahead and grab one of these, but we got a sponge last dive and I'm kind of holding out for a coral. Got a bunch of coral people on right now and I think they'd appreciate sort of holding off for a bit, seeing if we can get some really unique coral. See how this is attached to. So these are very small fibrous attachments, but it's still fibrous as you can see right underneath it. And that's quite informative. Yeah, so the family's definitely right. I'm quite certain it's in the Pheronematidae, but I just not sure after that what it is. Texture of this sponge looks a little bit different than some of the other members of the genus Poliopogon.
Really looks like the screen doors on my house at home.
Yeah, and you're actually seeing the skeletal parts here.
Exactly.
Yeah. And these are all made of silica, and that's what gives the glass sponges their names. So you've got, if you were to dissolve the living tissue away from these spicules or skeletal parts, then they would, and looked at them under a scope, they would look very transparent and glass-like, almost crystalline.
Okay, I'll get a quick establishing shot.
Yeah, thank you. That's a nice
Should we
shot of
turn
that
off the
one.
lasers for that?
Lasers off.
Gonna back off. I went to Poliopogon. I'm gonna back off on that one to family. It's not something I like to do.
All right. Clear.
I'm starting to move again, so you should have a bit of tether. Roger.
It's got a shape to it that's somewhat reminiscent of a different genus, Semperella, and that's why it's probably better to be cautious.
Oh, that's about it.
We have about 1 hour left. And co-pilot?
Did I hear that right? We have about an
Yep.
hour
Did you
left.
say you saw us start to move about 30 seconds ago or so?
Yeah, I think that's right.
Okay. That's about 4 minutes then.
Okay.
I wonder what that circular thing is.
Yeah. I don't know what that is. It's another one of these euretid sponges that we saw right on the landing site. That's the one underneath the slab. Nav, watch lead.
Go ahead, watch lead.
Yeah, so I wonder if we could maybe punch in a little bit longer ship moves just so we can get up and survey that flatter area that we're headed to.
Okay, you looking at kind of this area right here?
Yeah, that's correct.
Yep, we can take care of that.
Great, thank you very much.
Our ship is currently stopped, pilots, and they want to get over to that flat area. And
We can keep moving.
okay. Would you like
Flattening out. So.
Yeah, it is flattening out. Would you like a slightly longer ship move?
Sure.
Okay. How about 50 meters? Okay. And co-pilot, are you still okay with 035?
Whatever takes us toward that flat point.
He seems to be facing at 65, and according to high pack, that's going to be the direction to go, 60 degrees.
Okay. Sure, we can go further to starboard.
Okay.
I'll turn in that direction.
Let's look at 65-ish.
Okay, I'm bringing Sirius around to 65.
Oh, okay. Bridge, RV nav. Hey, we're going to get another ship move in. We're going to go 50 meters, 065 degrees,
Again, we're
0
not seeing a
decimal 2
significant
knots.
change in the substrate type.
Good copy.
The little Bathypathes, still a lot of these little two-pronged primnoid corals, little two-branched, I mean. Still lots of black corals on the bottom. These are these brown ones that we've seen all day.
Good copy.
Video switching out.
Thanks, Tara.
Another of the Dictyolo space sponges that we were just taking a look at a bit ago. Yeah, so
It's this little white one that's coming into the middle there.
yeah. Oh, and there's a larger Poliopogon in the back there. Poliopogon, however you pronounce it. It's that sort of globular sponge right at the top of the screen. Yeah, so we've seen some smaller ones. That's a little bit bigger colony. Very heavily manganese crusted, wouldn't you say?
Yep.
So,
Yeah, definitely.
so you said nobody's ever aged this particular seamount, is that
Not
right?
that I'm aware of, no. I don't think anybody sampled this even.
There's a shrimp in the water column. This is our first a larger shrimp. You gotta take a look at this guy. He's
Let's get the shrimp.
yeah, just even a snap zoom, just to.
That's a partial zoom.
You can go in tight.
Alright, that's full zoom.
I think he's got a longer rostrum. or
Too
not.
tight.
Yeah, that'll be fine. Thank you. So he didn't really have a long rostrum on
Yeah,
him.
he's went
That's
straight
great.
into you.
I always get these backwards.
Check him out.
Without the rostrum, it's
It's a race between you
Yeah,
and
full
on
wide.
your iPad and the chat room.
well. I think Chatwin will win here. Let's see.
You're still good. Nobody's
Still
chimed in
good?
yet.
Okay.
You're still winning.
Keep looking. We still have a chance here, right?
That's right. Maybe somebody else has a faster, newer iPad.
So I think the Aristiids are the ones without the rostrum, and the Benthasimus are the ones with the rostrum. I'm trying to figure that out right now. And by the rostrum, I'm talking about a spine that's sticking right out from the center of their heads.
Scott's asking about a different coral to the left.
Benthasimus doesn't. No, I think it's Benthasimus that doesn't have the spined, pronounced spine. Wait.
I'm not entirely sure which one Scott's
What's
looking
that?
at.
Oh, sorry. He was looking at a different coral?
Yeah, maybe this guy here.
You're looking at the black coral, Scott, or is it? Oh, this one by the lasers. That's what he's looking at. Yeah, maybe we could do a zoom on that. It might be a bamboo coral. And I think it is. Good eyes, Scott. I was in shrimp land there while this showed up.
Yeah, come in tight.
That's tight.
Right, if you have any more, go ahead.
And he's branching.
Nope, we're all the way in.
Off away from the
Yeah, he's
node there.
Yeah, internodal. Patty Fryer. Bamboo!
I think those last two O's are meant to be extra loud even.
Yeah. So you can see in the stock, you can see these, I guess they call them needles. These are specialized sclerites in bamboo corals. And what Scott and Asako are looking at right now, I think, is this arrangement of these little rods or needles inside the stalks, because that's informative. It's an excellent view, Scott, but we need an ID. We really need an ID. I'm tapping my foot right now, waiting. That's trying to prompt Scott France to go a little further than bamboo, because he specializes in this group of corals, the bamboos, along with Les Watling, who's not with us today.
No, he's not going to commit.
Give us a clade, Scott. Even a clade. No, he's not going to do it. Okay, I said it. I guess bamboo. I have to be happy with that.
That's right.
Scott almost wants to call it, but it's internodal, Scott. It has definitely got a lot of needles in that stock. Angled body wall sclerites. What he's looking at are the rods that are in an angular formation in the stalk of the polyp, which is similar to the genus he thinks it could be. That the problem is that the Acanella is, if I'm not mistaken, is a nodal brancher, which means the branches come out of the nodes.
Right on top of you, pilot. You're still okay,
Okay,
for a
that's
minute.
great. I think we got enough. It's whenever video's done.
Yeah, when you're good video.
Alright, I think we're good to go.
Come on up.
Scott's agreeing with you that it should be internodal.
He wants to say Acanella,
Right,
but he
full
won't
wide.
because of the internodal branching. So. The Candidella gigantea, or whatever it is, it's similar to Candidella gigantea.
That one?
No, this one here.
Oh, yeah.
Remember the Charlie Brown coral?
That's right.
Charlie Brown Christmas coral.
That's right.
So who knows? Maybe we'll get into a few more bamboos here as we're going to. I think I see another one on the bottom too. So the community may be changing, and I think I see our first Chrysogorgia too, right by the lasers to the left of the lasers. And it is.
So that's partial?
So yeah, we are getting a change in the community finally.
Come in tighter.
We're getting very close to the top of this little, first little bump on this ridge.
Yeah, and also Scott thought he saw Piranapathes there. I was wondering about that one as well, Scott. So
I think I have
you're
a
talking
little
about
more too.
the unbranched one that was almost like
that's
a
it.
plume. Yeah, and the squat lobster there is a Europtychus.
Ship is stopped. Just came to a stop.
Okay, I'm still moving and I'm right above you.
You'll move for another 4 minutes or so.
Those are very common
come on out.
crustaceans that like to live in this type of coral.
Is that a
something behind it?
Or all the way out?
Let's do a partial, get this guy behind it as I go over. Come on in, video.
right there?
Come in tight.
That guy?
Yeah, it's like some type of a polychaete worm, maybe. Yep. See if anyone will go more than that. Maybe a scale worm. I always default to scale worms, polynoids.
I'm max
this
aft
type of
on
thing,
you.
but
push
I
out.
don't know the
Yeah.
worms at all. Do we have somebody
Full wide.
get on the audio? Maybe. Not really sure. Oh, Scott, is that you? I can barely hear you.
Yeah, it's me. I will try to, hold on. Does this sound any better, Chris?
Yeah, that sounds better. Now I can hear you clearly.
Okay, I'll have to use the handset when I'm speaking.
All right, we have got a lot of black corals here on the bottom, Scott.
Yeah, this is a pretty incredible place. Sako has been mentioning, and I think you've been relaying it, that the Crinoid abundance is just remarkable, and there's a number of different black corals as well. So it's an interesting spot. It's interesting that so many of these Crinoids are so small. I'm not sure if that's an upper growth size or if that's telling us something else.
That's a partial.
I don't know. And, you know, in the background here, we're seeing unbranched
Max after you
bamboos,
again.
really small bamboos with big polyps on them. So we're definitely getting a change in this community from what we first saw when we landed.
Look at this.
Oh
one.
yeah,
I'm
there's
seeing
now we're
that.
starting
I
to
see
see.
what you mean.
Yeah, there's a big bamboo coming on screen right now, Scott. So yeah,
Giant thing over there.
you should see it any second.
And these black corals, these light tan ones that I thought the unbranched one was Parantipathes. I see a number of them are branched now. I suspect they're all the same thing. I don't know if that's the genus, but it's possible.
I don't know, Scott. I just, you know, I saw what you were looking at when you brought it, when you mentioned it, and I think there is, it could be Piranapathes. What we'll need to do is zoom in on one of those. I don't know. It just looked different than the other branched
We'll be done
ones that
moving here
you
in about
suggested were
30
either
seconds to a minute,
Heteropathes
as
or
far as
Schistopathes.
Sirius.
Okay, I'm still moving pretty
There
quickly.
may be one
Yeah.
coming into view
I'll
right
hold
now,
off on the next
Scott,
call then.
right next to the bamboo,
Sounds
to
like
the
they're
right
going to
of
want to
it.
see this stuff.
Yeah, I'm looking at the bamboo, and it's interesting because this bamboo has both internodal and nodal branching. Oh my gosh.
A lot of it's nodal, though. Well, no,
Yep.
there's
A lot of it is,
internodal.
but not all of it.
No, it's not. I've just, as I said that, I'm looking at an internodal.
Yep. Scratch my head.
So this is clearly different than the bamboo we just imaged a moment ago, correct?
Yeah, I
Max,
would
back
say
at you. If
absolutely.
we're still moving, we're
This
going to
is
need to go soon.
probably in the J clade. That's the one clade we know of that has species that branch from the node and other species that branch from the internode. So it's possible you could have some that do both. Just a guess. most of the polyps here seem closed up. The tentacles are retracted, so they don't look as flowery as the others we've been seeing, which suggests that something has disturbed this colony recently.
Yeah, I
and
think
the number
I'm
of anemones
stationary.
growing on the branches.
That's a hydrozoan, I think. It's attached to the branch of the coral. It's probably a dead part of the branch because there's not any polyps on it. So yeah, it's nice to see these.
Come on out.
I always like to see the change in the community, so it's kind of nice to see it's changing now.
All right, we're full wide.
Agreed, Chris.
It'd be more comfortable if you could pull ahead a bit.
Yep. Yeah, another one of these big bamboos coming up. I'm not sure if it's the same one or not, but
Okay, come on in.
You should have as much time as you need here.
In this case, they're going on opposite sides. There goes
So
the
that's a partial
flagpole
zoom.
theory.
yeah, just from the colony, Scott, this thing looked somewhat reminiscent of Jason Isis, but I can't see the branching yet on it.
That's what he said about the
Oh,
previous
Jason
one.
Isis. So I just looking at
That's
I
what
have
he said
to.
about the previous one.
Oh, the previous one.
I think.
Okay. Good. So I have to lean over a bit to see the chat room where I'm sitting here. So I hadn't seen that entry. That's good.
It's good to know we're in agreement.
Yeah, except that this one looks clearly internodal, though, Scott. This new one.
Should I get rid of the lasers?
So, and I was talking about this one
Come
here,
on in,
so
Vinny.
Lasers off.
I probably blew the idea on this one.
It's funny because as we were approaching it on the side, the thinness of some of the branches, I was going to suggest it was in the Isidella, but they are definitely internodal, so this is absolutely not Isidella. This is classic keratoisis. Very strong needles there projecting between the tentacles. Very thick, I mean, the white sclerites. And then you can see there's a number of, in some of these, very short little rod-like sclerites. Again, they're the white things that you see in the body of the polyp, and they seem to be aligned almost like a series of bricks.
Yeah, right at where the polyp connects to the branch. So,
Yep, and moving up. Yep.
right.
And that's a nice view.
So in the space of just a few minutes there, we had potentially 2-3 different types of bamboo corals just appear out of nowhere.
Yep, I agree, Chris.
Yep.
Is it alright to zoom out for a full body specimen at some point?
The top real quick.
As you said many times, this is the value of being able to get a good depth transect to see this transition. These ones look like they are really packed. The polyps are really packed with sclerites. You can see what I'm seeing on the right side. Some of these polyps, you're getting just the right angle of light to see that they're fairly dense and they're fairly numerous.
Yeah, I see what you mean.
Could be B-clade keratoisis.
B-clade, okay.
It's also very impressive. I just noticed a moment ago we could see the tip of some of these branches, and you can see the growing tip had a whole bunch of sclerites stuck up there as well, and presumably a new polyp would emerge from the top there. They do continue to bud additional polyps as they get larger.
Yeah, so they're preloading them with sclerites already before even the polyp appears or the tentacles appear. I saw what you were talking about.
Right, exactly. So they're in the Scenenkime as well, which is the tissue that joins the different polyps.
Oh, wow.
When you're good, we can
Okay,
get
yeah,
an establishing
when
shot.
video's good, we can go ahead and see if we can find some other different things. As we move across these flats, we're sort of right on the edge of the flats right now.
That we're full wide.
I'll back off. Big guy.
Yeah, he is.
Hey, pilot, after you get ahead here, I can go ahead
There
and
we
call
go.
in the next ship move. Get us up to that flat zone.
Yeah, I need to push ahead quite a bit.
Yeah, after you do that. But when you're ready, let me know.
Okay. Good video?
We're good. Thank you.
Big, huge, old Euretid, I think, behind that. Like very similar to sponge when we landed. So there's just a sprinkling of these around.
Go ahead, Bridge.
And that might be the little high point there, or right as we come over this rise, maybe we'll start to flatten out a little bit. Looks like it's sort of shoaling a little bit right up ahead. This curly Q thing looks like it's a
Copy
dead
that.
stock of an Iridogorgia. Be nice
Copy
to see
that.
a live one.
Thank you.
I'm pretty sure that's what it is, but it's long gone, I think.
Come on in, video.
Now might be a good time for that
Is that
heading
partial
change
zoom?
if he wants
That's
to.
what it is, all right.
What was that, co-pilot?
Now might be a good time for that heading change if he wants to.
Assuming
He doesn't
that's
know
what
yet.
it
It's just
is.
now starting to swing around. It's not quite time to move
Copy.
it.
I would agree.
Yeah, it's got some bumps on the stock there, though. It's a little strange looking. You know, I wonder if this could be a Radicipes, Scott. I don't know if you're
No,
seeing.
I think the bumps are where the branches were when it was alive.
Yeah, okay, that's it, of course.
Come in tight, video.
That's full zoom.
Little Crinoid is probably in the family Antenodon. Antenodontidae, excuse me.
Alright, when you're good, video.
Okay.
Copy that. You can go ahead and do that right now.
Alright, full wide.
Did you see that orange round object under your right toe?
The anemone?
Oh, we got
I
a
think
bunch of
that's what
stuff.
it
I
is,
think we
yeah.
got a forest up
Oh,
ahead.
it's not as red as it looked.
Yeah,
It's
so over to the
it's
left.
remarkable, Chris. Yeah, I was just... Sorry.
Go ahead.
No, go ahead, Scott.
Oh, I was just thinking about that Iridogorgia close-up. We were just looking at the skeleton there, and all the fuzzy stuff on it was hydrozoan, and how much hydrozoans there are down here when you look up close at surfaces. They coat so much that it's an amazing, the abundance of these other cnidarians down here that we don't really get to identify the species. They're so small.
Yeah, it seems like if you give them just the tiniest opening, they can colonize something. There was a hydrozoan on just one little patch of bare branch on, I think it was on one of the icidid corals, the first one we came to, and there was already a hydrozoan on it.
I think I see an asteroid possibly preying on, is that a coral up here on the left side?
I'm trying to, oh no, it's on a sponge. Yeah. And then we got some beautiful big colonies of bamboos as well. So we're...
Absolutely. Yep.
Come in on the
we could take a look at this sea star here.
So, Chris, just from my, just for your knowledge, it feels like there's a current coming out of my starboard side, so from the south maybe.
It's coming from the south?
Yeah, southeast.
Oh, southeast.
We're
Okay,
looking
thank you.
east.
So that's a partial zoom.
don't know if you copied that, Scott, but Josh, the pilot, was mentioning that he thinks the current is coming from the southeast. So again, it's that would come straight over the side of the ridge.
And if you have any more Zoom, go ahead.
So as we're looking at this sea star here on the sponge, I wonder if it's feeding on the sponge, and I see just behind it there's a downed bamboo coral colony that has most of the polyps missing, not all, but most. So I wonder if that was a previous victim of another sea star.
Yeah, it could be, and I'm wondering whether this almost looks like the pythonaster that Chris Maw had identified, and it was also on a sponge, as this one was. I'm trying to find my sea stars in the gallery here.
Yeah, one of the things I'm not certain that the sea stars that feed on the sponges will also feed on the corals. I believe there are different species that do that, so I don't think this would be the same one that would do both.
I'm looking
that could be an opening for Chris to correct us.
Yeah, I'm looking at the picture of pythonaster that we came across last year. We actually collected that sea star and sent it to the Smithsonian, and I think it could be another one of these. This was very exciting for Chris when we grabbed that first one. This is Chris Maw, who's one of the world's foremost experts on sea stars. He's based out of the Smithsonian. And this was a really good find in his mind to actually spot one of these. I think this is what we're looking at. We're looking at another pythonaster here. But I sure hope he calls in. I saw him very briefly in the chat earlier, so we're putting out the Chris Maw spotlight into the sky. It's reflecting off of the clouds right now.
Hopefully he heeds the bad signal.
Yeah, the Maw signal.
The Maw signal. Yeah.
It's pretty late there on the East Coast, though. I don't know how late he monitors this.
Come out partial video. Do we want to look at that downed bamboo or downed coral?
If he's up, he'll certainly call in about this animal, I'll bet. So, and if we don't hear him, then what is that?
Would you want to look at that one? They were asking if we wanted to look at the one in the background.
No, but there's a coral over to the right, just as you were right at the farthest extent to the right. Yeah, that fan right there. I wonder. Oh, it's another. It's a bamboo, but it could be something. Yeah, half dead bamboo.
Go bridge.
Okay.
Something came
It's
right
kind of a
here.
nice little spot here in the monument.
Copy, thank you.
So Rocco is wondering what the very small white sticks are. Are these the stylastrons that we were seeing earlier, or is this something else?
Maybe we'll see if we can find some of those. Scott, did you want to look at one of these plume-like black corals too? There's a couple of them right in front of us now. If you wanted to take a look and see if you think it's...
Yeah, I won't be able to give you an ID now. It's possible if we get a look, we can see how many what they call primary pinnules. So those are the small branches that are arising from that main stem. Are they in sets of two, or are there smaller ones in between the twos if there are four? That kind of thing.
I see.
This looks like the keratoisis, perhaps, that we were looking at earlier.
Yeah, it's definitely internodal and.
Yeah.
Okay, that should.
Yeah,
that
so
should work.
You notice on these black corals, Chris, they have that main axis that attaches to the bottom, and they have these side branches that are certain length, but then they look kind of thick and fuzzy, really close to the axis, and usually that means there's some other much smaller branches, again, called pinnules, that are arising. So there'd actually be 4 or 6 branches, even though it looks like there's only 2, those paired ones. So that could tell us something. And certainly Tina would be able to tell us if she were on.
And that would rule out Piranopathes, wouldn't it? Because Piranopathes has got the paired branches, doesn't it? The longer. Yeah. So this does not look like Piranopathes to me. So then we're back to the Heteropathes or the what was it? Heteropathes or Trissopathes, maybe.
Trissopathes or Hexapathes, right? Exactly. Yeah. So these look to me, I agree that there's too many little branchlets in there to be a Piranopathes.
Watch lead, Nav.
Roger, Nav, go ahead.
Hey, just want to give you a quick heads up. We have about 30 minutes left.
Okay, well, let's see if we can go through this bamboo, see if we can see how far it extends.
Okay.
All right, we need a good video.
All right.
Yeah, so we can put a ship move in then if you'd like.
Copy
The video's full wide.
Yeah, you're still pretty much right under me, so I should get out ahead first.
Yeah. When you're looking from the serious view from the other video feed, you can sort of see how it seems to really be crowded up on top of this rock and around it. It may not be
I'm
quite
looking at
as,
another
quite
65
as busy.
degrees.
Yeah, that's what I'm wondering too, whether we're just in a
Okay.
small patch or whether this whole
50
flat
meters
area
still good
might
with
have
you?
a lot of stuff on it. Hard to say.
Is there a little metallogorgia in
Bridge,
there? I
Robbie,
see
Nav.
there's a little rounded cliff almost right in the center now.
Yeah,
Or
pilot.
Hey,
probably
we're
on
ready
the
to get
right
another
for
ship
you.
move of
No,
50
it's right in the center
meters.
right now. Pilot, I don't know if you
0.65
copied
degrees,
that.
0.2
Whether or
knots.
not you, yeah, they're zooming it right now, Scott.
Great.
And the answer is
Good copy.
yeah.
That's a partial.
I'm not sure it's metallogorgia, but it's a chrysogorgia, the stock chrysogorgia. It looks like it's got that stair-step bare stock. Could be metallo though, but
Right.
it doesn't quite look right. I don't know what you think.
Yeah, so I agree. It's definitely not metallogorgia. The question is it chrysogorgia or pseudo metallogorgia? And I should know this because this was a species that was described by my student and I can never remember if it has a monopodial axis or this sympodial, which is this sort of zigzagging back and forth.
Do you have
This
any
is
more?
probably chrysogorgia, but I'm going to
Alright.
check it out while you guys continue.
Okay. Hey. Yeah, so what he's looking at there is this bare stem that you can see that's zoomed in on right now. And metallogorgia, that tends to be kind of a smoother stem. This has kind of got little branched in horns, and it's kind of angular, zigzagging down there, and that's the problem with that ID on it. So he's checking it out right now on his reference materials.
Okay. We need a good video.
All right.
Yeah, pseudochrysogorgia doesn't zigzag that much. So let's put this into chrysogorgia.
Okay. Thank you.
Do we have a ship moving? Okay.
Now we've got 30 minutes to find a corallium for Amy Baco Taylor. And she may not be watching. I saw that she was on earlier, so she could be watching. She may not be on the chat, but it's really late for her. So, but you never know. One could pop up right at the end of the dive.
So you guys said this is a beautiful stand here. It's relatively dense with these large fans and something we haven't seen earlier on the dive. And Jasper, I think you were saying in the serious view, this looks like it's just a local high and isn't going to continue. Is that correct?
Yeah, if you look at the serious view, it sort of looks like this thing you're looking at, this rock you're looking at, sort of drops back down a little bit behind what the ones you're seeing, and it's hard to see, but it doesn't look like I see quite as many in the rocks behind it. But maybe it's just a light effect.
Yeah, it's kind
I
of a.
I see a couple of them still, I think.
Yeah, it's kind of a ridge-shaped rock, Scott, and they're lined up along the top of the rock itself, and the rock is sort of right along the ridge line. You know, so they found this nice little high point right on the edge of the ridge here that they seem to be clustered on.
It's interesting to compare the size of these bamboo corals with all the primnoids that we were seeing earlier that were so large. And excuse me, the primnoids are really small. These are large, and I'm now kind of skeptical that was all a recent recruitment event. It seemed like it was extending over particular species of primnodes just naturally do not attain a very high for whatever reason.
Yeah. No, I think there is a bunch of things in there that were pretty similar. But do you still think that those two-branched primnoids, do you think those still could be a young version of them four-branched ones, or were the two-branched something unique?
Well, you know, we saw an awful lot of two-branched ones, didn't we? And that just seems to me too much of a coincidence to see that there are that many at that same growth stage.
Right.
So I suspect they may have been something different.
Right, so that's what you're referring to.
You know, I suggested a couple of names. I suggested a couple of names like the Narella alvinae, just to point out that there are some Narellas known that have that very sparse branching. So it's certainly possible that it's that genus. Don't know the species for certain.
I think there's a fish in the middle.
We've got another one of these eels out here. And this is another synaphobranchid eel.
That's a partial zoom.
Yeah, come in tight.
Yeah, I think the dorsal is quite a bit back on this one, so it'd be potentially that Synaptobranchus oregoni or Synaptobranchus brevidorsalis that we saw the other day.
Not a little
that,
bit.
Ken Sulak was talking about.
Yeah, go ahead back in.
Looks like in that direction there's still a number of other corals, like the ones we were looking at.
Yeah, maybe we could come back down and see if we can see some more of these big bamboo corals, get a better idea whether it was just that one little rock or whether or not they go on a little ways on this ridge. Because this ridge, of course, goes quite a bit further than what we're able to survey today.
Yeah.
So it's
Alright, let's come back down. Brighten this up.
yeah, there's a few things in the background there, but it does seem to have petered out a little bit. And we're right on the sort of local high. There's some stuff over to the left. A few colonies sticking up.
Of course.
Yeah, there's a bunch in the background there, so it's a nice little stand.
Pilot, are we looking back at what we were looking at earlier, or are we looking in a different direction?
We are heading back in that direction. We're not there yet. So we're looking more to the southeast.
Okay.
But the actual little
Right,
top
so we're
we
not
were
looking at the same colonies that we saw before, I don't think.
No, those were
just looking
back
at the
farther.
track.
The ones
Now
in the
we are
back,
probably
yeah, the ones
looking
in the
back, but to the
ones
left
in the
is
distance
new.
are where we were before.
What was that, Josh?
The ones in the distance?
Yeah.
That's where we were before.
Oh, is that where we were? So they are the same colonies? Okay.
Yeah. But these guys in front of us and off to the left are new.
Okay. So we're going to be pausing just for a moment. They're doing a bit of a watch shift, watch shuffle here. We're going to enjoy this nice little stand of big bamboo colonies here while they play musical chairs up front.
Yep.
The new team comes in, the other team is leaving.
The question is, when are you going to get to musically chair yourself into the front row?
I don't know if that's in the cards.
Okay, watch these pilot with you.
Roger, is that Chris up there? Or
Yeah.
is the pilot? Hi,
Yeah.
Chris.
Hey. Okay, so my understanding is we're just going to try to follow this line
Yeah,
here for as long as we can.
Yeah, let's play Follow the Bamboos.
Okay. We can do that.
Thank you.
Looks like there's a couple below you.
Yeah, we played that game before, right? You remember last year?
Oh, yeah. Yep. Got pretty good at
Chris, I think we just passed over a very small Iridogorgia, so I'm going to make a note of it in the event log.
Oh, okay.
A live one.
Alrighty, I missed that one. Okay.
It was really small.
All right, now you got me on the Iridogorgia, Han. I'm gonna ignore these big bamboos, and I'm looking for the Iridogorgia. Of course, the Iridogorgia would be a really nice find here. So
Yeah,
Scott's
it looked like a Magnus Borealis. It had quite a big spiral, but maybe only 2 loops.
Okay. There's more bamboos continuing on up to the right there. So tapered out a little bit, and we've got a fish here, and it's a different fish. I wonder if we could zoom this fish. I think it's our first macrurid.
Go ahead and video.
He's moving fast.
Oh, just hold there. See if we can at least get an ID on him.
Is it a macrurid? No.
It's a swim away.
Keep coming. Hold there. Yeah. Oh my goodness. Come on. Oh, hold there.
Yeah, he's a macrurid, all right.
Push back in. One more time. Hold there.
Well, the nice thing
Yes,
about
he
that
is.
fish is it's pointing out that there's also these small primnoids down here, along with these bamboos.
Yeah, I can see what you're
So
talking
thank you,
about right
fish.
behind him. Yeah.
Let's let him go.
So this little
Alright, come back
macrurid
away.
is what we were calling Kumba species in the
Alright,
Northwest
roll wide.
Hawaiian Islands and in the Marianas.
Thank you.
He was very active. He has no right swimming that fast in this cold of water. I don't know what he was up to there, but it was rude. He should have posed a little more.
Yeah.
Yeah, I can turn my head to line up with these corals.
Yeah, and I don't
Nav,
think
what
the
was the
lights
ship
on the
move,
ROV, they're all LED, so they don't give up a lot of heat.
Ship
either,
move
but-
is complete. Cirrus does a little. I think it just settled out now.
What was the heading on that?
065.
65. Okay.
Yeah, I can see them continuing in the background. You know, they taper out a bit, and then you see a bunch more. Not quite as dense as that little high spot that we were at, but they're still around.
Yeah, you can turn your head. You want to?
Chris, I'm also
Copy.
seeing a lot of
Copy
these
that.
sponge stocks without any head to them. They seemed alive. I mean, they're white. Have you talked about that already? And perhaps I didn't hear earlier.
Yeah, we did. So they seem to me that they're most likely Caulophacus stalks, and those can be white for quite some time. And I'm
What's up?
basing this on the fact that they don't really widen.
13
Like
minutes.
Bolosoma tend to
12 minutes.
widen, both at the base and toward the top. But we haven't seen a lot of Caulophacus down here today, too.
Got you at
so
33
I'm on
degrees.
the lookout for a live
Right.
one of those as well.
Mean.
Just to confirm
Copy
that.
that.
Like, here's a big stalk right here, and this is probably could be a big Bolosoma.
Maybe get
It's
one
a
last
dead
photo.
one, and this is
Yeah, we can go
brown.
ahead and do that. You want to?
What heading are you thinking?
Yeah, so a lot of dead stalks. I just don't know what that means.
Just
We're
continue
just,
on the
They're
065.
just hanging on, and they haven't fallen over
Maybe something
yet.
a little closer to 90.
We're just seeing
Uh,
the progression.
Yeah, maybe it drops off. I don't know.
birth, life, and death in the deep sea,
20
or what's going on.
25, 30 meters, 080, something like that.
Yeah, is this transitioning from a sponge community to this black coral, bamboo coral, Crinoid-dominated community? Interesting question.
Yeah, well, we had a fair number of black corals,
so 30 meters at 080
earlier down, but we also had a lot of sponges. And
go ahead, video.
There's less sponges up here so
but
far.
There's definitely a lot of, well, still a lot of black corals right where we're looking.
Yeah.
So on the map, it looks like we kind of crossed over the very summit part of this. And
co-pilot 080, 30 meters, 0.2 knots.
Sort of
yeah.
Cruise right behind where all these
bridges
big
and
fans were.
Yeah, so we're kind of coming down just slightly in a little bit. And
exactly.
They're preferring the little high knoll there.
They're really high, like sitting high up on top.
Yeah.
Bridges is Nav. I'd like to put a ship move in, 30 meters, bearing 080, 0.2 knots.
That's a bamboo down below here.
Putting in one last ship move, because I'm at the end of my tether now.
That's correct. Ship move underway.
Thank you, Nav.
Thanks, Nav.
Nav, we got about 10 minutes left on the bottom.
You're muted now.
Yeah, it's about all right.
So about 10 minutes left.
10 minutes left?
Yeah.
Okay. So we have about 10 minutes left. I don't know if Scott or Masako, if either of you see anything that might be worth grabbing right at the last moment here at the gun.
Copy that,
what you feel about these bamboos, Scott, whether you think it would be worthwhile grabbing a branch or so if there's enough time to do that, or whether these
I do. Either one. I think the one, if we were lucky enough to stumble on the one that had both the nodal and internodal branching, that would be, I think, the ideal one. But either one, we can't identify unless we have it. So, I would say either of those are targets if you come across one that's convenient for sampling.
Okay. Well, we've got a couple up ahead there. I don't know if we could make it there in time to be able to grab one really quickly before we leave bottom.
Yeah, we'd need to set up for that now.
Oh, you were? Okay.
No, we would need to if we're going to take one.
But to be fair, the black corals as well, they're certainly a dominant member of the community today. That would be something that could possibly be interesting.
Yeah, I guess the main thing is that
let us know,
Right. I think the main thing is we are running out of time, and this is a late dive just because of the little issue they had earlier. So we may wind up.
Yep, understood.
I think we could possibly nab something for you here.
Yeah, it's nothing really sticks out unless one of these black corals. You were saying, Scott, that one of these black corals would be informative?
Well, I'm saying that the black corals were really dominant today, this patch, and so that would be interesting. Asako is actually noting that the Crinoid recruits, almost anything that's in front of us right here would be of interest and value.
Did you copy that, pilot?
Meaning any of these small...
Yeah, see these small brown and white things are right where the lasers are? They're suggesting that...
You spun up here.
Absolutely.
Yeah, if you were able to get right over there and grab one of those things, that would...
Hydraulics ready.
That would be a nice final act.
Okay, I think we could squeeze one in here.
Just take the whole
Chris, I think I see right in the center right now for me, lower center, there's an unbranched
Nav, do we have any biological samples today?
Candidella gigantea. Never mind.
How about right
Negative.
Right above where the lasers are right now? That guy?
Or Yeah, would you prefer one of these branched Trisopathes, Heteropathes, or unbranched, Scott? Do you have a preference? Just trying to think of which would be easier for you with the manipulator. Pilot, maybe the one on the right.
We can get any of them
you
on
can
top
get any
here. On top of the...
Yeah, because I don't have a preference, I would say whatever the pilot feels is the easiest thing to sample given the short bottom time remaining would be useful.
Okay. Yeah.
All bio boxes are empty.
He says any of these would work for him. Any of these brown ones.
Okay.
Not the dead stuff, of course. Those light orangey.
Not
plume,
the one.
Yeah, the plume-like.
In the foreground to the right there.
No, that would. yeah, exactly.
I bet that would get the rinse bag quite dirty.
So this would be collection specimen 2 bio, I guess.
Okay. Can you maybe man my pilot cam? Just bring it over. And actually just bring it up on the main. I mean on your main. Alex on. You get a partial zoom video. Hold there. Okay, so which one of these in here will actually...
Yeah, really, any of these would be fine. Whatever's easiest for you to grab, I think.
Okay.
If you get this snip behind it, you might get both of them.
Oh, you mean the
one behind us has like two next to each other.
Yeah, just hold there. How about these?
Yeah,
Can I just
yeah, just go ahead and grab them. I think those will work.
Okay, copy that.
Copy that, Bridge.
Thinking starboard or port.
Come port. You come wide video.
Full wide.
Copy. Do we have room to extend?
I think so.
The drawer.
Nice, you got the hold fast on the one on the left.
Come down on the...
yeah, that's going to be a nice specimen.
Pilot's ship moves completed.
Copy.
I'm going to draw out just a little bit.
Okay.
Actually, what's even better is you got one unbranched and one branched.
Come in while we got it in frame video, just to check it out. Let me move it for you. And come wide. Just fill the frame with the whole sample. Yeah, there you go. Just so they know what they have.
I guess we're going to call this one single collection. They'll go in together. They were collected in one event, even if it's 2 colonies.
Okay, we're just extending the drawer now. Watch lead.
And you're going to go port inside, is that correct?
Port inner, yeah.
Okay.
I'm going to use the pilot cam to take a look at that wing real quick.
Copy. I can come up too. Why don't I just do that anyway?
Yeah,
so
put Trisopathes, Trisso pathes, or Heteropathes, which is hetero, and then pathes. Yep, that's it.
All the way out.
Copy.
Pilot cam, I'll get that on there for you.
Come wide video.
Full wide.
Copy.
I may have been wrong about saying one was unbranched. I think I noticed a very small branch when we had a close look at the claw there.
Yeah, I guess we'll find out when they come up. They didn't.
My wrist is hitting there, isn't it?
By the way, these are probably pretty floaty.
Did you copy that, pilot?
Yeah.
Okay.
Yeah. see how they do here.
Very slowly deciding to go into the box.
You want me to retract?
Making up their mind right now, Scott.
You're going to give it a nudge. Oh, nice. Very nice.
Yeah. And they were just encouraged to go into the box.
Please go into the box.
Nice.
There we go. Okay.
Yes, with a little encouragement, they're
Nicely
in the box.
Done,
Yep.
Nicely done.
Great collection. Thank you.
Package is secure.
Copy.
So Chris Osako is asking again about these white sticks. I think there's several right here. If we get one last zoom of the bottom before we leave. I don't know if you identified them earlier.
Yeah.
You just give me an
They look like little white thumps sticking up the bottom.
Make sure we're stoked.
Yeah, we did get a close-up of one, and I don't see any right in front of us. This is that stylosaur-looking thing, right? Look.
Right. Yeah. Okay, if you had a good look earlier, that's fine.
Well, it was caught when they were zooming in on something else. I noticed one of them in the background.
Take my camera back.
But right now, I don't see any in front of us. But we'll try to get a still grab of that Osako. I'll try to keep that in mind and send you a picture.
We got 1 minute to spare.
Copy that. Let's get one more look at this. What they're
still mice
talking about back there.
Yeah, they almost look like little thumbs that are sticking up off the bottom, so they're not real long, they're real kind of stubby.
Okay.
And I just don't see any right now.
Did we pass any right where we
No, I
were
don't
sampling?
see any right now either. I tend to notice them when we zoom in on something else, so...
You want to do a partial zoom video?
How about this? Right on the edge of the ledge here, there might be some.
Oh, yeah.
See these things, these little ones? Yeah, I think we got some, Scott. And maybe Osako spotted these on one of the other views. Yeah, that's it, I'm pretty sure, is what she was talking about.
Yeah, because when we were collecting there were a bunch. So, right, I think a bunch are there on the edge. So isn't it interesting that there's virtually no bamboo corals right here, and yet we had that great abundance
start to come in
just a
video?
short distance ago?
Yeah, very, very patchy. There's another final shot of the double Crinoid.
Swing arms are stowed, hydraulics secure.
So you don't think these are sponges, huh?
I don't know, actually, but I didn't think so when I first saw it zoomed in earlier. It didn't look like it.
Let's just come in on some of these. You want to keep coming? Oops, sorry. A little hard to tell from that distance, huh?
I could swear we had something like this last year, and I thought it was a sponge at first and got scolded about that ID from somebody. So let me see if I can, if we kept
I apologize if that was me.
No, I think it was one of the sponge experts. It might have been Konrad Tabachnik in Russia who might have said, that's no sponge.
They just don't feel like
Did we want to recover with 0 turns?
Yeah, I think I'm going to look in this unknown
you turn right
group that we had.
I guess
we keep a picture of one of these.
Yeah, I think we do.
I just didn't know if they wanted to purposely recover.
Yeah,
some of them are fairly wide.
Can't really remember
on a relative
what we
sense.
decided.
Others are quite thin and
we daisy chained it
they look
straight.
a little bit soft.
So.
Okay.
Yeah, so I'm waiting. This is. Yeah, this is what Osako, she mentioned in the chat, this is what she's talking about. I wonder if she knows what these things are. Because you think they could be a soft coral, an alcyanation of some kind, Scott?
Right. Well, right, yeah, exactly. So, something like an unbranched plexaurid or... I don't think it's an ellicellid of some kind, but I still think
here,
sponge is a possibility. In fact, I'm looking at one here now on the right side. It looks like it has like cladorized like spines coming out of it.
Do you want to? Chris, we gotta go. Unfortunately,
when I see it is
sure.
After
Thank you.
have already seen it in person.
Yeah, it's.
Well, have a look at the video later. That was certainly pretty good view.
Right. I'm going to have to go look at my emails and see if we, if it's the same thing maybe that I sent off last year to try to get an ID on. But yeah, I'm not seeing it in the gallery, so maybe because nobody knew what it was, we didn't include it. So if you hang on for a moment, Scott, we'll go ahead and do the post dive wrap up here.
Yep,
I know
no
it's
problem.
really late.
I'll stand by.
What are we doing? Coming around.
If you turn to
on
starboard.
cue, Lindsay is right here, showing up.
It's going to be as good as we're going to get, I think. Yeah. Nav, you can pull up your web GUI, and then hit ascent once we start going up.
Good copy, got it.
Great, good deal. Okay, my camera's maxed out.
Copy.
And catch you on the flip side.
Yep. I got a couple more bamboos as we're leaving the bottom here. Right on that boulder, some nice sized ones.
Yeah, it really seemed like they were lining up right on that edge.
Yep.
Still got your fish eye a little bit?
Yeah, because it's real late
push out a little.
for the East Coast
It's
folks.
1:30 in the morning.
Yeah, so we're going to blaze right through this.
Come up a little.
I can see more
Are we
colonies
going on to camera 3?
we're camera 3, is that right? Okay.
Just to see.
Stand by, Scott. They're
so.
trying to get switched over. It'll probably be 3.
All right. Very nice.
Copy that.
Ready to come up on your mark?
Okay. Yeah, let's continue up. I'll push. Keep a little push
set?
ahead on.
Yeah, okay.
I'll let you spool your.
Yeah.
Yeah.
Epoch 2 or 3?
We are clear for takeoff.
Got it. Looks like it's good.
All
okay,
aboard.
Right.
The Blue Roto
So, Scott,
Express.
can you see the map right now?
Not yet. Still got the quad view. Oh, there it is. Yep, it's up.
Great.
Okay, and do we have anyone else on the line right now? I see that Brendan Rourke is in the chat room. Hi, Brendan. I hope you're doing well, and I know that Asako's still here, so maybe we'll just go right into this. I'm pleasantly surprised by today's dive. Obviously, whenever we find a whole bunch of animals and see a different community, it always pleases me, and that's what we saw today. And even though we had a shorter dive, we did manage to get a rock and a nice biologic collection and cover some ground. So I don't know.
I'd say it's a credit to everybody, given the problems at the beginning of the dive, that we're able to get down on this site and see what we saw. I'm not used to seeing a community quite like that, that's so dominated by those small primnoids and black corals. That was an interesting thing to see.
30 meters
Yeah,
net.
very interesting. I agree. We hadn't seen a community
30,
like that before, so it's
31.
terrific. On your screen right now, you're seeing tomorrow's dive. So for those of you who are listening still, we're going to be diving every single day now all the way through the monument until we have to leave and transit to Kwajalein. So there are no more transit gaps that are going to occur. So I'm hoping that everybody will plan to be with us every single day for the next couple of weeks. Lindsay has got up the map for tomorrow's dive, and it's an unnamed geo seamount, as far as I know. Jasper, is that correct?
Sizable, yeah. It's respectable.